Hello, Dr. Deborah. How are you, madam?
Deborah, how are you, Deborah?
Hello, madam.
Good morning. I'm very well, thank you.
Welcome.
Madam, we are live. Can we start?
Yes, video start.
How are you, Deborah?
Hello, madam.
Good evening. Good morning.
I'm very good morning. I'm very well, thank you.
Welcome.
Madam, we are live. Can we start?
Yes, video start. Yes.
How are you, Deborah?
Hello, madam.
Good evening. Good morning.
Good morning. I'm very well, thank you.
Welcome.
Madam, we are live. Can we start?
Yes, video start. Yes.
How are you, Deborah?
Hello, madam.
Good morning. I'm very well, thank you.
Welcome.
We are live. Can we start?
Yes, video start. Yes.
How are you, Deborah?
Good morning. I'm very well, thank you.
Welcome.
We are live. Can we start?
Yes, video start. Yes.
How are you, Deborah?
Good morning. I'm very well, thank you.
We are live. Can we start?
Yes, video start. Yes.
How are you, Deborah?
Good morning. I'm very well, thank you.
We are live. Can we start?
Yes, video
Good morning. I'm very well, thank you.
We are live. Can we start?
Hello. Assalamu alaikum. I am Professor Samina Chowdhury, President, Endometriosis and Adenomyosis Society of Bangladesh. Greetings from Bangladesh. Good morning, good evening to all of you. Welcome to Endo Bangladesh 2021. My salam and respect to all of you. This is the first time in the history of Bangladesh, Endometriosis and Adenomyosis Society of Bangladesh is hosting the first international scientific conference today. In this conference, we have got world-renowned speakers, researchers, and faculties. They are 70 international speakers, researchers, academicians. They are international. And in the national one, 121 faculties are directly involved as speakers, chairpersons, moderators, and rapporteurs. 17 countries are involved in this conference. A total of 28 sessions. Already 5 sessions are completed as pre-conference on 8th of October, which is a very successful one. And rest 23 sessions are waiting in these 2 days. We'll complete it. Your contribution and participation, especially the viewers. I want to congratulate all of you that you are the key of the success of this conference. And the theme of the conference is Combat Endometriosis Together. I wish that we all of us work together to fight against endometriosis. Thank you very much. Please enjoy the session. And over to keynote lecture number 1, please. Nusrat Mahmood. Unmute yourself.
Dr. Lima. Thank you, Madam. Ladies and gentlemen, participants from home and abroad, good morning to you all. I am Dr. Lima Shompa, welcoming you to our first International Scientific Session on Endometriosis and Adenomyosis Society of Bangladesh. Our session is keynote lecture. We're really honored to have Professor Rohana Hathatoa and Professor Samina Choudhury Madam as our chairperson. Our moderator of this session is Dr. Nusrat Mahmood. She is Associate Professor in Infertility Unit of Obs and Gynae Department of Bardem Hospital. Our chairperson, Rohana Hathatoa, he is President SAFOB, Secretary General of AOFOG, immediate past Chairman, Ministerial Disorder Committee of FIGO, President World Justice Organization, past President Sri Lanka College of Obstetrician and Gynecologists, past President Menopause Society of Sri Lanka, Vice President SAFOM, and Treasurer Asia Pacific Society of Infective in Gynecology and Obstetrician from 2010. Our second chairperson, Professor Samina Choudhury Madam. She is President, Endometriosis and Adenomyosis Society of Bangladesh, immediate past President, Obstetrics and Gynecology Society of Bangladesh, Chairman, EASB Hospital and IRCH, Chairman, Endometriosis Subcommittee from 2018 to 2020, Member, FIGO Committee for Human Rights and Refugee and Violence Against Women. Now I am over to Dr. Nusrat Mahmood Madam. Thank you, Madam.
Thank you, Dr. Lima, for your nice introduction. Knowing the fact that endometriosis is a common gynecological condition in childbearing age, yet there is diagnostic dilemma. Our first speaker will be talking on the burden of endometriosis in COVID-19. He has vast experience in managing the endometriosis patient all over the world, all over the country and in the South Asian region. He is none other than Professor T.A. Choudhury. Professor Choudhury is the recipient of Shadhinota Padak, the highest national award honored by the government of Bangladesh for his outstanding contribution in medical education and women's health in Bangladesh. He also received the Gorkha Dakshin Bahu Award, the highest civilian award from the government of Nepal for their contribution in making medical specialists for their country. Former president of South Asian Federation of Obstetrics and Gynecology, Bangladesh College of Physicians and Surgeons, former vice president of Asia Oceanary Federation of Obstetrics and Gynecology. Currently, the honorary chief consultant and professor of a reputed medical college and tertiary research hospital in Bangladesh. May I request Professor T.A. Choudhury, sir, to deliver his talk. Over to you.
Sir, unmute yourself, please, sir. Yes, sir.
Chairpersons, first of all, a sort of just to tell you that this presentation was prepared when the COVID-19 was nearly up to its peak. Fortunately, it has got a trend to go down now, and many countries are nearly free from this scourge of this century. But in any case, I think we'll give a presentation on this topic. Unfortunately, there are many patients who had COVID also with endometriosis, but literature is not rich on this topic. So I'll just talk with whatever is available now. So before I talk, I think I will give a little overview of both endometriosis and COVID-19. And most of you probably will be knowing this, but in spite of that, I think it's better to start the discussion from that point. Now, you all know what is endometriosis. So it is an estrogen-dependent chronic inflammatory condition which is manifested by endometrium-like tissue outside its normal location, that is the uterine lining. The important thing is that this ectopic tissue, they respond both to estrogenic and progesterogenic stimulation, like normal menstruation. And during each menstruation, these deposits bleed, and they elicit a inflammatory response, and it goes on gradually expanding the lesion wherever it is in the peritoneum or in the rectovaginal septum. Next one. So, what is the extent of the problem? Endometriosis can affect any woman of childbearing age. Different surveys have shown that the incidence is about 10 to 12% among women of reproductive age. But if you consider women of certain special groups, those suffering with chronic pain in the abdomen, or those who are suffering from infertility, the incidence may be as high as 40%. And we all know that endometriosis has got a big impact, not only on the person who suffers from this disease, severe dysmenorrhea, dyspareunia, pain, etc. But in spite of this, there is a long delay in diagnosis, generally from 8 to 11 years. And societal impact is also there because of medical costs, surgical cost, caregiver cost, and so on. So it is a huge burden on the society also. WHO has summarized more or less the situation of endometriosis. They say 1 in 10 women suffer from endometriosis, maybe total 170 million women with endometriosis suffer from severe disability or difficulty during their reproductive age. And most of whom have not been diagnosed easily. So progression, as I have said, that the mechanism, presence of endometrium-like tissue outside the body, the collection of menstrual debris produces acute inflammatory reaction leading to release of cytokines, ultimately adhesions and cyst formation. Presence of the ectopic tissue evolves an estrogen-dependent chronic inflammatory process. Thus, endometrium is a cellular, focal, and local disease, but can ultimately involve large areas of the body in some cases. Now, there is mostly this is a condition which deteriorates with time, progresses with time. But Lancet in 2019 said that some of them may remain static, and there is a small possibility that some of them may actually regress even without any treatment because of the natural resistance the body develops against the progression of the disease. Next. Now, why endometriosis occur? And this is also a riddle because it was first described by Daniel Schoen in 1690. And we are still in the same situation because we are not very sure about its definitive origin. So during last 3 decades, there has been lots of research, and we understand the clinical aspect and the treatment aspect of the disease somewhat better. but we still do not know what actually caused the endometriosis. But it is more and more cases are being diagnosed now. It may be due to actual increase in lifestyle, or it may also be due to better diagnostic modalities by which we can diagnose this disease better now than before. So endometriosis is considered a difficult disease to diagnose and treat because it has got lack of specific symptom, poorly understood pathogenesis, limited therapeutic option, lack of predictability regarding progression or regression, and the treatment may have profound effect on patient's fertility status. It is also strongly associated with the psychological makeup of the individual. Sites of endometriosis, I need not go into details. You all know what are the possible sites. It may be within the abdominal cavity or maybe remote from the source. But one thing I want to emphasize that endometriosis can occur in lungs and thorax in a significant subset of population. And since COVID-19 primarily affects the lungs, what would be the effect if a patient who has got thoracic endometriosis also gets COVID-19? It's not yet very much clear, but we'll talk about it a little later also. So pathogenesis, there are lots of things. Simpson's original theory was retrograde menstruation, which still is valid in many cases. Subsequently, Mullerian transformation was considered to be a possibility and can explain some cases of endometriosis. So these metaplastic changes still can explain some cases. But recently, newer concept has come. One was of infection. Endometriosis was associated with certain types of infection of the genital tract. Then, and last is the stem cell theory, where it is postulated that stem cells which remain dormant, either in the genital tract or sometimes even in remote location, like bone marrow, can ultimately settle and can be transformed into endometrial tissue. So these are all theories. No one is absolutely clear which causes endometriosis. So this is schematic representation that how the possible theories of origin of the endometriosis. Next. Next. So clinical presentation, you all know, I need not go into details. They are variable and non-specific. That is the problem. So unless somebody has got a high index of suspicion, they will not be able to diagnose endometriosis. And that is why usually there is a delay of many years before a definitive diagnosis can be made. Next. Now, some facts about COVID-19. You all know this is a highly infectious respiratory coronavirus, which was first detected in China and then rapidly spread all over the world. WHO declared it as a global pandemic on 11 March 2020. So though most of the cases were mild, it produced severe illness in 16% of the patients with a high mortality and post-illness morbidity. By the end of September 2021, it has been estimated that more than 150 million people have been affected by COVID and more than 5 million people have already died. But those who have died, apart from them, there are many patients who had severe hypoxia and had to be admitted in the hospital. And that produces a severe shortage of oxygen and ICU beds, which has not been seen any time before in the last century. So measures taken by the different government to combat this health problem So in some cases, lockdown of the whole country or the country was in part of the country was enforced. Public transport was suspended. Shops and offices are closed. Many people lost their job. So there was a serious economic problem for many families. So on the other hand, endometriosis is a very painful disease. It produces high level of stress, both physical and mental, and impact many aspects of life such as education, work, social life, and marital relationship. Many such patients need frequent medical attention, either for adjustment of their medicine or waiting for their scheduled surgery. COVID-19 pandemic has changed all that. So the government has imposed curfews and quarantine measures have been enforced in many countries. This has reduced family and social interaction, increasing mental stress. Stay-at-home measures have impaired availability of physician consultation prescription, and many cases surgery has to be postponed. And there was also a sense of uncertainty among these patients that when they'll be able to go back to their regular follow-up or their scheduled surgery. Next. COVID-19 patient and endometriosis. There is no report that patients with endometriosis are more likely to become infected by COVID-19 virus. A small subset of endometriosis patients, as we have told before, have thoracic endometriosis. Though there is a possibility that these people may be more vulnerable, there is exactly very little information in the literature to say whether this is true or not. But I think there are some patients in endometriosis who rapidly deteriorated. And these are the cases who might have thoracic endometriosis, but nobody is sure. Next. So effect of enforced public health measure on the health service as a whole, not on endometriosis only. So there was suspicion of OPD services. Only emergency services were maintained as most health workers were relocated to provide service to COVID patients. There is lack of availability of service which produce severe anxiety in patients who are suffering from diseases like endometriosis. Planned cold surgery has deferred, and even they were anxious that when the services resumed, how long it will take to clear the backlog before somebody gets a chance to get the treatment. So it produced both physical problems as well as severe psychological problems. Next. So stress is a big problem. Apart from the actual pathogenesis of COVID-19, the stress of uncertainty about when and where they will be able to get their treatment produces severe psychological breakdown in many endometriotic patients who contracted COVID at the same time. And this is still persisting because in many hospitals it is not possible to clear the backlog even though services have been resumed. Next. So are endometriosis patients more vulnerable? We have discussed it, that up till now there is no evidence that they are more vulnerable, or once infected, they are more likely to have serious disease. As you have said, there's a small subset of thoracic endometriosis which may have a different prognosis. Next. So effect of COVID-19 infection on patients suffering from endometriosis, just summarize: a reduction of the quality of life due to delay in diagnosis and treatment, temporary closure of OPD services, postponement of planned surgery, and fertility treatment, and longer waiting list once the service resumes. But how long it will take depends on these resources available at that particular facility. In Western countries, they will be able to get over probably better than many of less fortunate and less resourceful countries. So the advice that was given that if an endometriosis patient gets COVID-19, so just to reduce the load on the health service, the advices that were given is that the initial consultation should be the family physician or a local gynecologist rather than a specialist. More emphasis should be given on clinical diagnosis and management to avoid complicated and time-consuming investigations. Take more use of telemedicine for referral and follow-up, and referral should be done only for emergency cases. All recommended health measures should be enforced during person-to-person contact. Next, there is one very good study from Puerto Rico which attends many of these interaction between endometriosis and COVID-19. Next. So they have shown that the effect of COVID on patients' lifestyle, behavior, and everything, there had been a change. Next. So during the pandemic, in that study, 53% of the subjects reported needing medical attention for their endometriosis symptom. 46% reported difficulties scheduling an appointment, with majority being for gynecological consultation. So of note, 23% did not seek medical attention due to fear of contracting COVID-19. So the problem was there. Even service was available in a skeletal form. Many people are afraid to go for that service. Medication commonly used by participants were found to be over-the-counter drugs, analgesics, NSAIDs, and sometimes occasionally prescription drugs. But that was generally advised to avoid if possible without medical attention. Next. So this is the cloud analysis of endometriosis. Next. So what is symptoms reported as worsening in endometriosis patient during COVID-19? So fatigue and back pain, headache. These are the main post-COVID symptoms in endometriosis patient, but this was not unique. This was possibly for all patients. Early in the pandemic, it was found that 14 to 20% of the endometriotic patients were actually deteriorated. Use of analgesics increased by about 25%. But fortunately, these are all over-the-counter drugs, not any drug having serious side effect. Next. So effect of COVID-19 on endometriosis. COVID-19, we have discussed, it is a sort of repetition, just to say that it has dominated the whole lifestyle of all patients, including endometriosis patient, and this is especially important in diseases like endometriosis, which requires frequent medical consultation and as well as readjustment and adjustment of that drug from time to time. So they are psychologically disturbed when they get a pandemic like this. Next, not many papers are available. I have given a few lists. You can run through it quickly. If somebody is interested, they can look into it. Next. Next. The monitoring of endometriotic patients. monitoring the impact of natural disease such as pandemics on health and well-being is a public health priority. So it has proven to affect pain in intensity and quality of life of endometriosis patients. So, but unfortunately, longitudinal studies with long period of follow-up is needed in order to find out actual magnitude and duration of this adverse event, so that in future, possibly we'll get more information on this topic. So burden, to summarize, burden of COVID-19 on endometriosis, worsening of endometriosis symptom, changes in risk behavior, substantial barrier in access to medical appointment, scheduled procedures, and prescriptions. Electronic health modalities considered as acceptable alternative for gynecological consultation in such natural disasters, which appears from time to time in all societies. Next. So this is the advice summary. Unfortunately, this was supposed to be projected at a larger screen, but in this small screen, I don't think you can see it very well. But these are the treatment options which are given to the patient, what to do in an emergency and future consideration. Move to the next one. This is too busy. Now, one thing here we want to say that the benefit of electronic health platforms, e-modalities, health modalities such as applications and telemedicine as an alternative solution has to this type of disease has now emerged, and improving accessibility to medical specialists has increased because of this electronic media and telemedicine. This is probably one of the benefits that this COVID-19 infection, people have accepted that telemedicine is recognized and reliable method of consultation, and all patients do not need person-to-person contact. Next. So conclusion, COVID-19 pandemic imposed an increased burden on health and the mental well-being of endometriosis patients. Many barriers have been seen in access of health-related services, as we have described. Patients considered e-health modalities as an acceptable alternative to minimize these obstacles. Follow-up studies to capture longitudinal data correlating such pandemics or even epidemics and its effect on endometriosis is warranted, as it will be important to assess whether those with endometriosis are more vulnerable to COVID-19 or they had a more severe type of disease. In the long run, alternative plans for care of patients needing long follow-up, like those suffering from endometriosis in crisis period, should be prepared. Thanks for your attention.
Thank you very much, sir, for elaborating us how to maintain the new norms of our life, that is COVID-19 with managing patient with endometriosis. There are scientific controversies regarding the etiology of endometriosis. To add to the note, our next speaker, Professor Caroline Garget, will be talking on the new concept of etiology of endometriosis. Professor Caroline is the Senior Research Fellow of National Health and Medical Research Council, Deputy Director of Ricci Center and Head of the Endometrial Stem Cell Biology Laboratory at the Hudson Institute of Medical Research, received several international awards for her discovery of endometrial stem progenitor cells. Former President of Australian Society of Stem Cell Research and Secretary for the Society of Reproductive Biology. Serves on the editorial board of reports. Floor is yours, Madam. Professor Caroline Garget.
Madam, just unmute yourself. Yes.
Okay, is that visible?
Yes, ma'am.
Oh, good, thank you. So thank you very much for the invitation to come and talk to you on this really most important occasion for the first conference for the EASB. And I do congratulate you on the program that you've put together. I'm going to talk to you about new concepts in etiology of endometriosis. So what I want to talk about is a bit about retrograde menstruation. And we know that this is sort of one of the most common theories of its cause, and we've just heard about that in the previous talk. However, retrograde menstruation occurs in most women and only 10% develop endometriosis. So it's got a multifactorial etiology. And what I want to talk to you today about is endometrial stem progenitor cells in retrograde menstruation. And then I'll also talk about some new knowledge in the area of genetics, in the endometriosis risk genes, somatic mutations, epigenetics, that may lead to, has given us new insight into the causes of endometriosis. And then there's a lot of interaction between the genetics and environmental factors. But before I start on the endometrial stem cells, I really want to talk to you a little bit about really new information on the structure of the endometrium, and in particular, the functionalis and the basalis. I'm just going to get my laser pointer. So here's just a typical picture of endometrium in the proliferative stage. And you can see down here in the basalis, the branching glands. And, you know, this is how we've always depicted it in our schematics for many years. But new knowledge, just recently published by Darren Happengama's group, has shown that basalis glands are really very branchy and very interconnected. And this has also been shown by the Enomoto group in Japan, who've done a tissue clearing and based 3D imaging of full thickness endometrium. And they too have shown this network of glands down in the basalis, shown here in yellow, and they've described as a rhizomal network of glands. And what they've also shown is that several gland can grow or emanate from individual rhizomes in that basalis region. Now, this has really important implications for menstruation, repair, and endometrial regeneration. And also for the, because I think the enzymes that break down endometrium break down the extracellular matrix. But when they get down here to the basalis, it's really stopped, I think, because there's the density and complexity of the glandular structure. Now, the location of the stem progenitor cells might be implicated in this too. And what Darren Happengama showed is that a gland growing vertically can actually comprise of two different rhizomal structures from the basalis region. I want you to remember this image here of a gland profile of two different parts, because I'll show you some more of that later on. And I think this will have implications in the pathogenesis of endometriosis. So now let's go back to the stem progenitor cells. And we first found these in the endometrium as clonogenic cells. So individual cells could grow up a clone of endometrial cells. They're really quite rare, as you can see here. Then we characterized them for stem cell properties. They could self-renew, we showed in a serial cloning strategy. They had high proliferative potential, like a single cell could produce several billion cells and undergo 30 odd population doublings. And then we took individual clones and we could culture them in a 3D culture, and the epithelial ones would produce this large gland-like structure. And the stromal clones established by a single cell could be made to differentiate down a number of mesodermal lineages. So we concluded that human endometrium contains rare epithelial progenitor cells and mesenchymal stem cells. Now, this information, this tells us about endometrium having functional stem cells present, but it tells you nothing about the identity of the cells or even where they're located. So we set about then to try and determine what their identity was. And with the endometrial mesenchymal stem cells, we tried a candidate approach, and we used a variety of markers. And here I'm showing you these two markers, and this is a flow cytometry plot of endometrial stromal cells. You can see this small population here, and it's this population that contained all the clonogenic cells. So we used that CFU or clonogenicity assay to determine whether the markers we were testing could show us functional stem cell activity. And then when we looked in endometrium, we could see these yellow cells co-expressing those two markers were right around blood vessels and in fact pericytes. But we also wanted to have a single marker. So we then screened with a small panel of perivascular markers, and the one that produced the best results for the clonogenic assays and all the other MSC assays was SUST2. And here you can see down in the basalis of endometrium, for example, the red staining or yellow staining where there's colocalization around veins and around arterioles of that SUST2 marker. So using my old-fashioned schematic, this shows us that the cells are perivascular around blood vessels, and they're around vessels in the basalis where we expected to find them, but also up in the functionalis. And this is actually important. Firstly, it meant you can obtain these cells by a biopsy. And secondly, for endometriosis, it means that they're shed into menstrual fluid. And I think that's a really important observation. So to look at the epithelial progenitors and find their identity, we did an unbiased gene profiling approach. And we took reproductive age endometrium and postmenopausal endometrium, and we got the highly purified epithelial cells from these, and we compared them by gene microarray. And our reasoning was that firstly, postmenopausal endometrium might be like basalis, and secondly, we know that if postmenopausal endometrium sees estrogen, it regrows. So it must contain some stem progenitor cells to initiate that regrowth. We looked for surface markers, and then we needed to find surface markers upregulated in the postmenopausal, or what we thought was the basalis endometrium. And the most consistent one here is the CDH2 gene. Now this encodes for N-cadherin, and here we do our clonogenicity assay, and you can see we get large and many more clones for the N-cadherin positive cells, and they also produce large gland-like structures, they serially clone, and they are highly proliferative. So when we looked in endometrium to see where they were indeed in the basalis, as one would expect with that experimental design. But you can see they're right down here against the myometrium in these branching glands. We looked to see whether we wanted to know, because N-cadherin is an epithelial mesenchymal transition marker, did they co-express with E-cadherin, which is present throughout the endometrial glands, but we found co-localization right down there in the bases of those branching glands. So we don't think it's EMT. We also looked for estrogen receptor alpha, and they do express estrogen receptor. And you can see this gland here. The N-cadherin is only half of that branching gland down there in the basalis. And more interestingly, when we looked at SSCA1, it's another basalis epithelial marker that's found by Darren Happen-Gama together with our N-cadherin, we showed that they really didn't co-localize. But again, we got those half gland profiles, half with N-cadherin and half with SSCA1. So I've redrawn the schematic, and we think we found that an epithelial hierarchy in human endometrium with the most primitive and clonogenic cells, the N-cadherin positive, SSCA1 negative cells right down in the basalis on one of those rhizomes. There's a very small amount of double expressing, and then there's these SSCA1 cells, and they go up a little bit into the functionalis, we believe. And then there's all these grey cells which don't express either of those two markers. But you'll notice that the luminal epithelium expresses SSCA1. And we believe this is because when the endometrium is repairing during menstruation, that the SSCA1 is migrating out of the stumps of the glands that are left and to resurface that epithelium. Then as the endometrium grows, the SSCA1 cells down from that basalis reach the luminal epithelium. So having identified the two endometrial stem progenitor cell types with functional activity, we wanted to test our two stem progenitor cell hypothesis in relation to Sampson's. And so our hypothesis was that the epithelial progenitors and the EMSCs are shed into the pelvic cavity by retrograde menstruation, and where they establish lesions in susceptible women. Now, to do this study, we did an investigation of women having laparoscopy on day 2 of their menstruation. And here I'm showing you graphs of that menstrual fluid that we collected actually out of the uterine lumen because they were under anaesthetic. And we've reported them in cells per mil. And this is a log scale. And we found no difference between control and endometriosis in the shedding of these the N-cadherin positive cells into menstrual fluid. And similarly with the SUST2 EMSCs, we found no difference. And these are present in a much higher concentration than N-cadherin cells. But when we looked in peritoneal fluid, far fewer of these endometrial stem progenitor cells were on a linear scale now. And the medians for control is 0 per mil. And for endometriosis is 195 with quite a bit of spread. And similarly for the SUST2 positive cells, the median for control is 0. And for endometriosis is 230 per mil with this spread of result of data. This is only an apparent increase of N-cadherin epithelial progenitors and SUST2 EMSC in the peritoneal fluid of menstruating women with endometriosis compared to controls, because our study, unfortunately, was underpowered. This was just very difficult to do, and we really did want to have more samples. So, having sort of established that Sampson's theory can apply, that the stem progenitor cells can get into the pelvic cavity, and they have got the capacity as cells of origin to initiate lesions. So now I want to just talk a little bit about the genetics of endometriosis, because we know it's a hereditary disorder and that it runs in families, and twin studies have shown a heritability of 50%, but it's a complex inheritance and it involves many genes, so it's a polygenic disorder. And I'm going to just talk in a minute about the common germline variants account for about 26% of endometriosis risk. So what are these genetic variants? These are single nucleotide variations, or SNPs, that have been identified by these genome-wide association studies. And these look at these very large studies have now discovered 40 genomic regions that are robustly associated with endometriosis. And some of them are shown down here on this schematic of the individual chromosomes. These are extremely large studies. And for example, this one has got, tens of thousands of cases and hundreds of thousands of controls. There's a greater risk, a greater percentage of risk factor genes in women with severe endometriosis compared to mild. Now, as a polygenic order, these many SNVs contribute just a small amount to endometriosis risk. They're found in the non-coding areas of the genome, and this means they're involved in gene regulation, and they influence the transcriptional output of genes in that particular region. And I just want to give you a couple of examples. For example, there's two risk variants that are located very near the estrogen receptor alpha gene, okay? And they're also in the same region as SNVs that have been associated with breast, ovarian, and endometrial cancer. Some of the SNVs are estrogen regulated, like GREB1. There's a FSH-B, that some of them are proliferation genes and angiogenesis genes. All of these, you can see, could be involved in the pathogenesis of endometriosis. Now, the thing about GWAS, it just tells you about variants in the genome, and they have been related to endometriosis. So it's important then to be able to understand whether their role in actual endometriosis. And so fine expression mapping of the SNVs in endometrial tissue is really important. And some of these have been validated in tissue, but others have not. So candidate genes from different genomic regions are involved in linked pathways associated with estrogen-mediated endometrial cellular responses. But there's a big need now to determine the functional effects of these SNVs in specific endometrial cell types. Now I want to go to somatic mutations, and this is particularly in the epithelium of the endometrium, so in the glands. And these have been found in eutopic and endometriotic epithelium, so in the lesions. These are from genome-wide exome sequencing, so they're in the coding DNA of the purified epithelium. They use laser-captured epithelium. They arise because of DNA replication errors and environmental effects that damage the DNA. Now, non-synonymous SNVs, these are ones that change the protein composition or structure. They've been now found in eutopic endometrium of women with endometriosis, in all kinds of lesions, and in endometrium of normal fertile women. The somatic mutation rate is quite similar to common malignancies, and some of them are in cancer driver genes, and these have been identified in lesions but also in normal endometrium. And in particular, KRAS and PIK3CA, PI3 kinase gene. The mean allele frequencies are quite low. They're usually around 20% or less. And some of them, like KRAS, the most common, can be quite high. So the clinical phenotype and the functional significance of these somatic mutations still needs to be determined. But I did just quickly want to go through one study by Suda et al., who looked in the individual glands of endometrium and showed clonality of those glands and a certain set of somatic mutations, and also in endometriomas. So each gland had its own unique set, and the lesions shared those same eutopic mutations, but they had their own mutations as well, which sort of suggests they've progressed. Some of the ovarian cancer driver mutations, as I've mentioned before, KRAS and PIK3CA were found in both glands and the endometrioma, but ARID1A was only found in an endometrioma. Lesions from multiple sites had similar mutations to the eutopic glands, which really suggests the seeding of them. And there was increased mean allele frequency for KRAS in particular in the endometriomas. Now, the PI3 kinase gene mutations will give a gain of function to those endometrial epithelial cells. It'll give them a proliferative advantage. They can take over the clonal niches in the glands themselves. And if they get into the pelvic cavity, they've got a growth advantage and they can escape senescence and cell death. And you can see how this could set up a lesion. So retrograde menstruation of endometrial epithelial cells with cancer-associated mutations leads to this selective advantage at the ectopic site and the development of endometriosis. And there's really a two-cell hypothesis here too, because these somatic mutations are in the epithelium. And there's also the niche cells, the stromal cells, that can be involved. And a very recent study has actually found some mutations in them, but no cancer ones, no cancer associated. Lastly, I want to just talk about the epigenetics, the other part of the genome, which really is affected by the microenvironment. And this is the alteration of the chromatin conformation. These epigenetic modifications open up the chromatin to allow transcription factors to initiate gene transcription. So they alter DNA accessibility to a modulator, which is the transcription factors. These are often enzyme mediated. So there's the DNA itself, which can be methylated or unmethylated by enzyme action. The histones, which are the proteins that allow that chromatin to be packaged so tightly, can be opened and closed according to the methylation status. And there are then these small microRNAs, which I'm not going to talk about today. And factors that can affect and influence epigenetic changes are like local estrogen production, inflammatory cytokines, and hypoxic stress. I just want to give you one quick study on this. Again, it's a genome-wide epigenetic modifications. This has been done in the stromal cells and cultured stromal cells, and they did a DNA methylation profile, and they found altered gene expression and progesterone resistance. When they compared endometriotic versus normal endometrial stroma, there were 403 differentially regulated methylated genes. Some of these were important transcription factors and nuclear receptors that are involved in endometrial stromal function. Many genes were involved in decidulization and altered steroid metabolism, inflammation, and endometriosis. So retrograde shedding of these epigenetic abnormal stromal populations may establish the stromal compartment of endometriotic lesions. I just want to give you one quick study to show how this epigenetics works. And this is looking at these master regulators, these two GATA genes. GATA2 is normally hypomethylated, so it's transcribing, and it's involved in genes involved in the decidual response and endometrial maintenance. And GATA6 is repressed, it's methylated. Now, when you have in endometriosis stromal cells, this GATA6 has been hypomethylated, and the GATA2 has been methylated and switched off. And then there's a whole lot of genes that are abnormally regulated that are involved in progesterone resistance, steroid metabolism, and estrogen receptor genes. And these then alter the and induce estrogen biosynthesis and progesterone resistance. And so you can see how this can influence gene expression and involving the infertility aspects. So just to summarize the whole talk on new concepts in the etiology of endometriosis, we've developed new methods to study the rare endometrial epithelial progenitors and EMSC. We've identified them and we've shown that they can get into the pelvic cavity, and they have the capacity, because they're clonogenic cells, to actually establish lesions. Sequencing technologies have made a huge difference because they've increased our understanding on the hereditary and on the environmental contributions to endometriosis. The GWAS has identified quite a lot of SNVs, risk variants, and we're studying them now in endometrial stem progenitor cells and functional studies to elucidate their role. There's a two-cell hypothesis of endometriosis development. The somatic mutation, well, this is what I want to bring to you, bringing all of this together. Somatic mutations and or the variant SNVs alter utopic endometrial epithelial cells, including, I think, the stem progenitor cells, and the epigenetic modifiers alter the utopic stromal cells, and no doubt the EMSCs as well, that differentiate into the decidulized cells. And this, we think, is a role then in the infertility associated with endometriosis. So the shedding of utopic endometrial stem progenitor cells with altered DNA and gene regulation may confer an ability for those cells to initiate endometriosis in those susceptible women, that thereby we've been refining now Sampson's theory of retrograde menstruation. So finally, I need to thank people in my group who contributed to the study I've talked about today, my collaborators and the funding bodies who have helped us undertake this work. And I'd like to thank you for your time and attention. And I can stop.
Fantastic talk. Thank you very much, madam. It's interesting to know the genetic influence and the environmental effect and their epigenetic expression in endometriosis patient. Thank you very much for your time. Dear esteemed audience, we have two distinguished personalities to chair our session. Dr. Rohana Hatatua, he is the president of South Asian Federation of Obstetrics and Gynecology. May I request Mr. President to give a remark on as chairperson's note. Dr. Rohana, please.
Thank you, Nusrat, for that kind introduction and request to speak.
Firstly, I would like to congratulate the Endometriosis and Adenomyosis Society of Bangladesh for organizing this really wonderful and informative international Congress involving so many international speakers and those local speakers with so many subjects being covered. So I also like to thank Professor Samina Chowdhury and Professor Saliha and for giving me an opportunity to participate in this Congress and give my comments in this first session. This first session, we were very fortunate to have really two learned, very renowned speakers. Of course, we all know of Professor T.A. Chowdhury. I mean, he is the most recognized and most respected teacher in this region. And we were very fortunate to listen to him. And he very well outlined and showed us the gravity of this problem, the endometriosis. As he was mentioning, though from 1600s, I think, this was being recognized and so much of investigation and research is being done, but still we are searching for the etiology, the pathogenesis. And also we are not really we have not understood the correct diagnosis and the management. So still it is in evolving, but it is an increasing problem as and it is affecting the quality of the life and also the economy and the family and the social well-being also. So it is a problem which we are all facing. And also Professor Chowdhury very nicely elaborated on how COVID-19 had its effects on endometriosis. Fortunately, there is, as he said, there is not much happening to these patients with COVID-19, and they are not very much affected. And also the disease is not worsened. And even if they develop COVID-19, they don't get much complication. So it is nice to hear. But the management has been affected by COVID-19. So it was a very illustrative, very informative lecture. So we're thankful to Professor Chowdhury for delivering. And then, of course, we had Professor Caroline, who took us to the very real cutting edge of this endometriosis, and she enlightened us on the recent developments. And as Professor Chowdhury said, so still we are trying to find out the etiology. So actually, so we are fortunate to have Professor Caroline, who is working on this edge, and she showed us how the stem cells and genetics, epigenetics, all are being studied and how they are contributing to this etiology of endometriosis. So there are so many new findings which have been developed, and so we were very fortunate to listen to Professor Caroline, who really enlightened us on these new developments which she is working on and which is taking place around the world regarding the stem cells and the genetics and the epigenetics. So thank you very much to both the speakers, and thank you for the opportunity given to me. Thank you.
Thank you, sir. May I now request Professor Samina Chowdhury, President of Endometrial and Adenomyosis Society of Bangladesh, to make her remark.
Thank you,
Madam,
Dr.
please.
Nusrat Mahmood, for your wonderful moderation. And I feel honored with chairing with Rohana Hatatua, and thank you very much for the kind words to me. And first of all, I want to say something about the T.A. Chowdhury sir, whose presentation, and it's very difficult for me because he is my teacher and he is my mentor. And I feel it difficult to comment about his presentation. However, I feel honored that in one word, I can say that it's a fantastic presentation. And he mentioned about the burden and its effect on endometriosis in the COVID era. Already there is diagnostic delay in endometriosis, and this COVID era made it worst condition and prolonged it, and disease is progressing, it continues. Sir, given us the guideline how to combat the situation. Thank you, sir, again. And about Caroline Gargett, what I say, I don't know, excellent and very thought-provoking presentation. And she is a great researcher working in this field to find out the etiology of endometriosis since the last 15 years. And I feel proud to comment on this presentation. She mentioned that the etiology is multifactorial, and etiology involves endometrial stem cell, progenitor cell, and genetic factors such as endometriosis risk, genes, and somatic mutation, and epigenetics, mesenchymal stem cells of endometrial vasalis and endometrial functionalis may mediate the angiogenesis and the stromal regeneration. Thus, endometrial stems and the progenitor cell may contribute to the pathogenesis of endometriosis by the retrograde shedding after menarche and from the neonatal uterine bleeding. Thus, the endometrial mesenchymal cell stem cell may promote the angiogenesis during the endometriotic lesion development. Fantastic presentation again. Thank you. Madam, for your presence, and please, your representation will encourage many of our researchers to work with you. Thank you very much for coming again, and we hope to meet again. Thank you, Caroline. Thank you very much.
Thank you, madam, for your nice remark. We are running short of time. I would request my co-host, Dr. Lima, to wrap up the session. Dr. Lima, please.
Thank you, madam. It is my pleasure to thank the honorable chairpersons. My thanks to the speaker for their excellent deliberation, which has enriched our knowledge enormously. I'd like to thank our moderator for her nice moderation. Last of all, I'd like to thank Novista Pharmaceuticals for their technical support. Now I am welcoming you to our next plenary session on this hall, and thank you to all, and happy good day to you all. Thank you.
Over to the next session. Farzana Rahman Rinki and Dr. Farzana Diba. Let's start the session. Dr. Rinki, unmute yourself, please.
Okay. এখন শোনা যাচ্ছে? Ladies and gentlemen, participants from home and abroad, good day to you all. I'm Dr. Farzana Rahman, rapporteur of this session, welcoming you all to the first virtual international scientific conference of Endometriosis and Adenomyosis Society of Bangladesh. Our session is the plenary one. We are honored to have Professor Roshanara Begum and Professor Firdausi Begum as chairpersons. Professor Roshanara Begum is the counselor of Bangladesh College of Physicians and Surgeons, past president of Obstetric and Gynecological Society of Bangladesh, professor and head of the Department of Obs and Gynae, Holy Family Red Crescent Medical College. Professor Firdausi Begum is the president of Obstetric and Gynecological Society of Bangladesh, past president of SAPOG, member technical subcommittee for SRH, Women's and Children Health, World Health Organization, professor department of Obs and Gynae, Ibrahim Medical College and Bardem Hospital, Dhaka. Now, the moderator of this session is Dr. Farzana Diba, associate professor Reproductive Endocrinology and Infertility Department, Bangabandhu Sheikh Mujib Medical University. Dear audience, in this session, we have eminent speakers, Professor Khaled Khan and Professor Deborah Bush. So, the session starts now. I am handovering it to Dr. Farzana Diba.
Assalamu alaikum. Thank you, Dr. Pinky. Dear audience, in this session, as Dr. Pinky mentioned, we have two eminent speakers, Professor Khaled Khan and Professor Deborah Bush. The speaker will have 20 minutes for the presentation. The speakers are requested to complete their presentation within 20 minutes. Before 2 minutes of end of the presentation, they will have a reminder. And for the audience, please, if you have any queries, put it in the chat box. At the end of session, we will discuss the queries if the time permits. It's my pleasure to introduce Professor Khaled Khan, our first speaker. Professor Khaled Khan is the Associate Professor of the Department of Obstetrics and Gynecology, Kyoto. Professor Khaled Khan is the Associate Professor, Department of Obstetrics and Gynecology, Kyoto Prefectural University of Medicine, Project Director, International Academic Exchange Center, and Chief Advisor, the Clinical and Translational Research Center. He is the Ambassador, World Endometriosis Society and Asian Society of Endometriosis and Adenomyosis. Now, may I request Professor Khaled Khan, sir, for his presentation. He will speak on current status, development, and problem of adolescent endometriosis. Professor Khaled Khan, sir.
Okay, thank you. Thank you so much for your kind introduction. So it is my great pleasure to give a talk in this outstanding meeting. First of all, I want to thank Professor Samina Chaudhuri, Professor Saleha, and especially Professor Sharmin Abbasi for their hard work and great effort until now to make this outstanding meeting very successful. And I feel proud of you very much. So can you see my slides? Not yet. Can you see my slide?
Yes, sir, it's working. Yes.
Okay, it's working. Okay. I want to give you some information on our most recent findings on the development of adolescent endometriosis. First of all, I want to talk that what is the current status of adolescent endometriosis. So this is the annual report from Japan. As you can see here, in 1997, the prevalence of the adolescent endometriosis is 58 per 100,000 population. And in 2014, this prevalence goes down to almost half, to 29 cases per 100,000 population. So what is the reason for this difference? Because in 1997, most of the diagnosis based on the symptoms, but after the 20 years, the diagnosis based on mainly the ultrasonography. So this is a study from Belgium, and they found that among 1,000 study population, the frequency of adolescent endometriosis is 0.9%. We performed the retrospective study in Japan in a single institution. The Saiseikai, the Nagasaki University Hospital, this is our collaboration partner. and we retrospectively searched the cases of adolescent endometriosis from the medical records of the laparoscopic surgery. So here is the result. We found that during the period of 2009 and 2015, among 3,455 cases, the total endometriosis cases was 1,270, about the 37%. And among these 1,270 cases, the prevalence of adolescent endometriosis was 18. That means only 1.4%. And among these 18 cases, 11 cases that belong to the stage 1 endometriosis, and the remaining 7 cases belong to stage 3-4 endometriosis. So we should know about the morphological appearance of the adolescent endometriosis cases. Here you can see that we should know that most of the lesions in adolescent women are the hemorrhagic lesions, as we can see here that in stage 2, 1-2 endometriosis and stage 3-4 endometriosis, most of the peritoneal lesions are composed of the red lesions. And I want to tell you what is the difference in the ovarian endometrioma in the adolescent endometriosis women. I didn't show you the slides. We found by the contact laparoscopy that the mucosa of the adolescent ovarian endometrioma is highly vascular with less distribution of fibrosis. In contrast, the mucosa of the ovarian endometrioma cyst wall in the adult women is less angiogenic and mostly mixed with the fibrotic tissue. This indicates the progressive phenotype of the ovarian endometrioma. So now I want to talk about the current development of the adolescent endometriosis. So in addition to the adult endometriosis, Dr. Sampson's retrograde menstruation hypothesis and Dr. Myers, the coelomic metaplasia theory, is equally involved in adolescent endometriosis. Recently, there is a controversial hypothesis, and according to this hypothesis, it says that neonatal uterine bleeding is associated with the early onset endometriosis. And this study comes from the Belgium, from the department of Professor Brozens, and he's hypothesized that the stem cells and progenitor cells do exist in the neonatal uterine blood, but it is just a hypothesis. There is no evidence. So according to his hypothesis, he mentioned that in the neonatal uterine blood, these stem cell containing neonatal uterine blood, when it goes back to the pelvis, it remains viable until the onset of the thelarchy or the menarche. And with the onset of the estrogenic action, these stem cells and progenitor cells proliferate, and they are the main cause of early onset endometriosis or the adolescent endometriosis. And according to his report, neonatal uterine bleeding affects only 3 to 5% of newborn girls, and it occurs 3 to 7 days after birth. And there are some high risk factors. So according to the report of Professor Brozens, he said that post-maturity and preeclampsia are the high risk factor for the neonatal uterine bleeding, and the prematurity is the low risk factor for the NUB. So we try to reconfirm this hypothesis that is neonatal uterine bleeding really occurs or not. So we tried to perform a retrospective search of the obstetrical medical records from 2013 to 2017, and we found that, yes, neonatal uterine bleeding indeed occurs. As you can see here that among 1,700 newborn babies, 807 are the female newborns, and among 807 cases, neonatal uterine bleeding occurs in 25 cases with a prevalence rate of 3.21%. And our prevalence rate coincides with the previously published reports as shown in this slide. And we found that neonatal uterine bleeding occurs 4 to 5 days after birth. And this neonatal uterine bleeding continued only 1 day in most cases. And then we searched all the maternal characteristics and also the neonatal characteristics as shown here. And then what we did a multivariate logistic regression analysis using one dependent variable, that is neonatal uterine bleeding, and 6, the independent variable as shown here. And we found that prolonged gestational week, that means long gestational week, 39 weeks or the more than 39 weeks, were significantly associated with the occurrence of neonatal uterine bleeding. And we also found that younger age of the mother was also significantly associated with the occurrence of neonatal uterine bleeding. That means, unlike a previous study, we found that longer gestational week and younger age of the mother are the high risk factor for the neonatal uterine bleeding. And also, as shown here, that other comorbid disease of the mother, such as intestinal disease, atopic dermatitis, the non-assuring fetal status, and hydronephrosis, these comorbid diseases are also significantly associated with the occurrence of neonatal uterine bleeding. So now the question is neonatal uterine bleeding is truly associated with the symptoms related to endometriosis? Then what we did, we went back to the 20 years before to search all the medical records who are now younger women, either adolescent or young women. So we searched all the medical obstetrical records from 1996 to 2000. That means these newborn babies are now grown up and they are now young women. And we found that during this period, the prevalence of neonatal uterine bleeding was 9.7%, about 3.3 times higher than the current occurrence of neonatal uterine bleeding. And we found that the gestational week at birth is significantly longer, and a maternal age was significantly younger in 1996 to 2000 cases than in 2013 to 2017 cases. And then what we did an online questionnaire survey because we have a very nice registration system. We have the telephone number of all the cases. So this was a very hard job. So my PhD students, they worked hard to have a telephone contact with each and every case who are now young ladies. And among 1,800 female newborns, we explained them in detail about the research protocol. And finally, we could retrieve the online questionnaire survey response from the 31 cases with neonatal uterine bleeding and 52 cases without neonatal uterine bleeding. So this is the demographic profiles of young women who were born with and without NUB in 1996 to 2000. As we can see here, the NUB cases are significantly younger than NUB negative cases, and the maternal gestational age at birth was significantly longer than NUB negative cases. So we just shortly summarize the response of the questionnaire survey. As you can see here there, do you feel pain during cyclic pain, during the period? Do you feel constant pain, acyclic pain, and the degree of cyclic pain, degree of acyclic pain? And does your cyclic and acyclic pain disturb your daily life activity? Do you need to absent from school or work during pain? Do you take any painkiller or the hormonal therapy for pain? Interestingly, we found that the degree of the severe cyclic pain and non-cyclic, there is no difference in NAB positive cases and NAB negative cases. That means NAB positive cases who are now adult, they equally complain of symptoms which are related to endometriosis. And there are other symptoms like cyclic and acyclic pain disturbing daily life activity, or absent from school or work, or they need any painkiller or the hormonal therapy. Found that they also complain, the similar complaint of these symptoms which are related to endometriosis. Now this is our ongoing study. We are prospectively collecting neonatal uterine blood to search the stem cells and progenitor cells in the neonatal uterine blood. But before that, what we did, we studied the neonatal endometrium collected from the autopsy cases. So here you can see the hormonal reaction. We collected neonatal endometrium for 15 cases. Here you can see here. And so this is the neonatal uterus. So you should know that the corpus is shorter and the cervix is very long. And in the neonatal uterus, this cervix is blocked by the mucus plug. So not all the neonatal uterine blood, it can't come out, but most of the uterine blood goes back to the pelvis. And here you can see the endocervix, ectocervix, fornix, and the vagina. First, we tried to see the response of the estrogen and progesterone. We found that estrogen and progesterone receptor were equally expressed as in adult endometrium. And then we tried to see the decidual reaction by immunohistochemical analysis of two decidual marker. One is the prolactin and another is IGF-BP1. And we found that 9 out of 15 cases and 1 out of 15 cases were immunoreactive to these decidual markers. And we also checked one pre-decidual marker, that is the glycodelin A. We found that 10 out of 15 cases that do express in the gland cells and partly in the stromal cells in the neonatal endometrium, similar to the adult endometrium. But there is some interesting report that this glycodelin A has a very strong immunosuppressive effect on cancer cells, other human tissue, and also in the immune cell. So then now the question is there any association between higher expression of glycodelin A and decreased infiltration of the immune cells? So we found that the CD45 expressing panleukocytes and CD68 expressing a macrophage infiltration was very low comparing to the adult endometrium. And here is the result. Comparing to the neonatal myometrium, these two immune cells infiltration was significantly low in the neonatal endometrium because neonatal endometrium is very thin comparing to the adult endometrium. So we combine the data between endometrium and myometrium, and we found that numbers of CD45 and CD68 cells in the combined neonatal endometria and myometria were significantly less than in adult endometria. And we found a weakly negative correlation between the expression of glycodyline A and CD68 and CD45 positive immune cells in neonatal endometria. So what does this result mean? It does mean that if glycodyline A expressing cells in the neonatal uterine blood goes back to the pelvis, it induces a potent immunosuppression. So these cells are easily survive until the onset of thelarchy or the menarche, and with the onset of estrogenic action, because these cells has estrogen progesterone receptor, they're easy to proliferate, and maybe this is one of the mechanistic basis of early onset endometriosis. So this is the part of our data of our ongoing study. We are trying to, as Professor Caroline, she already mentioned about the progenitor cell stem cells. We found that CD99 and CD1052 mesenchymal stem cell phenotype we could able to identify in the adult endometrium and also in the adult uterus and in the positive tissue. So now we are trying to check whether the neonatal endometrium or the neutral uterine blood, they contain any stem cells or the progenitor cells. So this is the last part of my talk, the problems of endometriosis. Endometriosis has a big problem for the women's health, as you already know. So how big is the problem? The problem is it causes the severe pain and ultimately cause the decreased quality of life. And if we ignore the early onset endometriosis, it will progress and it will induce an inflammatory reaction in the endometrium and pelvis, and ultimately it may contribute to a state of subfertility or infertility. And another big problem, it is not mentioned here, that even we do the sophisticated medical management or the surgical treatment, there is obvious recurrence. And one more problem is that if we ignore the ovarian endometrioma in adolescent endometriosis women, it will grows up, and part of this ovarian endometrioma undergoes malignant transformation. So this is the data from Japan about the malignancy rate from the chocolate cyst is 0.72%. And another big problem, as you already know, that the diagnostic delay, as you can see here, that in the adolescent women, the time interval between the onset of symptom and the final diagnosis is the 10 to 12 years. So this is a very big problem. So we need to motivate all these young ladies, young women, to come to the OBGYN department, so take a diagnostic investigation and proper management. So with this concept, we started a multinational and multi-center study that is the endometriosis promotion project in Asia. Our purpose is for early detection and early management of endometriosis in young women. And I'm very glad to say that many universities in Japan and other Asian countries, including Bangladesh, are participating in this study. So I want to thank Professor Samina and Professor Sharmin Abbasi for the great contribution for this study. So this study is still ongoing. So this is the take-home message from my talk. Number one, the information on current status of adolescent endometriosis is limited. We need further study. Number two, we confirm that NUB indeed occurs with a prevalence of 3.1% that coincides with the previously published reports. Number three, gestational age over 39 weeks and younger maternal age were significant risk factors for NUB. Number four, our questionnaire survey indicated that younger women who were born with neonatal uterine bleeding equally suffer from symptoms related to endometriosis comparing to cases without NUB. Number 5, higher expression of glycodyline A and decreased number of immune cells in neonatal endometria may explain the survival of cells in neonatal uterine bleeding after retrograde entry into the pelvis. And number 6, our ongoing study with neonatal uterine blood and neonatal endometrium may further clarify a possible link between neonatal uterine bleeding and early onset endometriosis. And if we can establish this very fascinating and new hypothesis in adolescent endometriosis women, then we can recommend the national registry system to input all the cases of neonatal uterine bleeding in the obstetrical records. So finally, I want to thank all my collaborators, doctors, and my good friends for their outstanding contribution to this time-consuming study. And I also want to thank all young women who kindly responded to the questionnaire survey, and also to all doctors and nurses who are kindly helping in the collection of neonatal uterine blood. So finally, I want to announce you that as an ambassador of Asian Society of Endometriosis and Adenomyosis, as you can see on the background image, we have 9th Asian Endometriosis Conference in Sri Lanka. Unfortunately, it is a virtual, the October 28 to October 30. So I would appreciate if you can register and participate in this conference. We have a very exciting and very attractive program. Thank you so much for your kind attention.
Thank you very much, sir. Thank you for your excellent deliberation. Now, may I request our second speaker, Professor Deborah Bush. Professor Deborah Bush is the founder and consultant Endometriosis New Zealand, Director EPP Coaching and Consulting, Director World Endometriosis Society, Principal World Endometriosis Organization. Professor Deborah Bush will speak on menstrual health and endometriosis, education in secondary school for early recognition and timely intervention in young people. May I request Professor Deborah Bush for her presentation.
Thank you very, very much for that very kind introduction. And I would like to thank the organizing committee for inviting me to speak at this very prestigious conference in Bangladesh. Thank you so much. And the presentations so far have been very, very enlightening. Professor Khan, I think my presentation will follow on from your presentation very nicely. So thank you so much for giving an introduction to this highly important topic. So I'd like to introduce you to the ME program. ME, of course, stands for Menstrual Health and Endometriosis. It's a secondary school education program that's been running in New Zealand, parts of New Zealand, for 22 years. One of the things that I recognized last century was that the difference between anecdotal evidence in the presentation of young women to the scientific knowledge that we had at the time. So of saying what we were seeing anecdotally was that symptoms do start young, was that young people with dysmenorrhea often have endometriosis, and that this was a much bigger problem in our society than we initially thought. So I'd like to bring this to you now, and let's get underway. So a disclosure, I do own the IP of the ME program, but I've not received royalties or license fee. I'm going to put to all of you to think about this take on a sonnet by Elizabeth Browning in relation to those young people who present with dysmenorrhea and possible endometriosis. How do we fail thee? Let me count the ways. Now, we've all heard these things from right around the world, the impact on lifestyle. And just as I look at the slide, I don't want to read all of the things that women have said, but I do want you to cast your eye over it, because whether we're in New Zealand, Bangladesh, the UK, or anywhere in the world, these are the things that we hear so often. 20 years to diagnosis, we heard that earlier in the presentation. The impact of daily life stops me from doing normal things. But I also started listening back last century to what doctors are saying, and sadly, some still are. So let's hear about that. You'll be okay when you have a baby. Women are designed to have pain. It's a woman's lot. I've never seen endometriosis in Māori women before. It's all in your head. You're too young to have endometriosis. You need a hysterectomy. Actually, it's a career woman's disease affecting white middle-class women. If you're a catastrophizer, a hypochondriac, a drug seeker, try this, try that, and come back in 12 months and let's see how you're getting on. She's an attention seeker. It's normal to have pain. You've had your children. You don't need your reproductive organs anymore. And what about this one? Endometriosis does not affect teenage girls. And that's the one really that started me thinking that was wrong and we needed to do something about it. And my approach was to do something about it in a very practical way. And I'm so thrilled that many of our esteemed scientists here attending this meeting are doing something from their perspective. This was mine. Back in 1997 to 99, we considered several things: what was in the medical literature, what was in the educational curriculum, and how were we going to implement a menstrual health program in schools. Here's some early resources that we had at the time, all about me, and we had a website as well. Things have moved on since then, though. So we planned, we designed, we held teen focus groups, we had engaged teachers. I set up some medical advisors, and we evaluated and evaluated and evaluated. The biggest problem, of course, was endo what? I mean, why would we take a program like this into schools when they'd never heard of endometriosis or dysmenorrhea? They'd of course heard of period pain, but that was just period pain, wasn't it? And so that was really the biggest thing we had, but we persevered, we tested, we evaluated, and we did an update, and we did a pilot study in a reasonably spread out region of small, but nonetheless spread out region of New Zealand. Always at the back of our mind and in our presentations, we were ensured we were being medically responsible and following educational trends. And in 2017, all those years later, the ME program is now published in ANZJOG in 2017. And what that publication showed was endometriosis education in schools, looking at the model, examining the impact of the disease, and actually if we were seeing earlier presentation of symptoms in these young people in our clinics. And this was a program that we ran 2 years ago. It was #CareAboutMe. In 2012, the World Health Organization published this report on how to plan a health education approach. And when we looked at that some 12 years earlier, we were really pleased that we followed these protocols and standards. Nonetheless, it is an absolutely planned approach to health education. And I thought I would draw that to your attention so that it may be helpful in the things that you do in your planning. Our aim, of course, was to report on audit data of the ME program and assess it for efficacy, and also observe whether it impacted on earlier presentation of symptoms. So the design was a well health educational program based on those WHO principles. We included data from the ME program, and then also retrospective data from a tertiary referral centre for endometriosis using mixed methodology. I want to draw your attention to this because these are some of the regions of New Zealand where the ME program went during that time of the study. And what we found was that in our study, similar to the one that you did, Professor Khan, was that we looked at some of these regions, and we said, how many of these young people were missing school every almost months with severe dysmenorrhea? So not endometriosis. And one of those regions was 37%. Mostly it came out to an average of 27% of our youngsters missing school every almost months with severe dysmenorrhea. Then in 2020 and 21, we had a look at this data again. The cohort was much smaller because, of course, with COVID, we weren't able to get into all the schools. Nonetheless, it still showed an average of 31% of our young people missing school every almost months. When I say young people, I'm referring to 15 to 18-year-old students. One of the important aspects we bring out in the program is the difference between menstrual discomfort and menstrual distress, where discomfort can be normal, distress with a menstruation is never normal. And we explain that in simple terms so that students can understand and be responsible for their health-seeking behaviours. This is data now from my own private clinic at Oxford Women's Health in Christchurch, New Zealand. So this is through my EP coaching, so endometriosis and pelvic pain, Well Health for Wellbeing coaching service. And I did a longitudinal study from 2001 to 2015. And this is just a bunch of earthquakes where I just couldn't keep track of data. So excuse the gap. But you will follow this trend line of the under 20 age group here and follow the trend line, and you will see the rise in presentation of symptoms, a little bit of a dip there, the gap, and then we go on so that at the end of 2015, 62% of the patients, 62% of the patients I was seeing were diagnosed now with endometriosis. And this peak here in 2006, even though it's now old, it's still relevant to what we're seeing today. Remember, please, as I present this data, that this is a tertiary referral centre. It is a biased cohort. But 96%, and frequently these young people had already not done so well. They've got the symptoms recurring and persisting despite being on the pill and other, might be JDES or JDEL. So 96% of them went on for laparoscopic review. 98% were diagnosed with endometriosis and half had stage 1 disease. It will be of no surprise to you to look at the presentation of symptoms, dysmenorrhea, lower back pain, bowel symptoms, bloating and fatigue. And in 2019, that had risen now to 62% of the patients who referred to me for this clinic are under 20, diagnosed with endometriosis. I want to introduce you now to a young woman called Raiha Paki. She's a Māori woman. She's speaking in Māori, but there are English subtitles underneath. We use this as a module in the ME program for our Indigenous Māori students, or for actually all of our students, to recognise to make it more approachable to them. So here's Raiha.
Ko Raiha Paki tōku ingoa, ko au te tehi o ngā kaipapa ho Māori ia o te aroa nei. I tēnei tautonu i mahi au I te tehi pūrongo e pāna ki tēnei mate, ara te mate kiri kōpū. He nui ngā akoranga I tau mai ki runga I au, I runga I taku pīkau I tērā kaupapa. Ko ku mōhiotanga o te ao o te wahine no o ku kuia no o ku whaia. He nui te mana o te wahine, he nui te mana o ngā whare tāngata o te ao. I tihi wā he nui ke atu pia I tō te tāne, he o ia no tērā, kōrero tērā. I mua I te taenga mai o te Pākehā. Ka reo tātou tūpuna I whakamā ki te kōrero o te whare tangata, te rerenga o te awa o te atua, te ai, te moetahi, te whakawhānau pepe, ka hore he whakamā o te kōrero moera mea. Hea take o te whakamā karekau, he mea nui te whakapapa, ko tātou a hine mā, ngā kai kawe I te whakapapa. Tēnei mate, te mate kiri kōpū ai. He mate mamai, he mate nui, whakararu ana I a tātou. Ki te moa tātou whai I te tehi huarahi e ora ai tō tinana, ko te pai ngā tū tēnā. He mea weri, te whakapā atu, ki te tehi mātanga, mō te tehi wāhi o tō tinana e tapu ana. Ai te mōhio au, he oi anō ki te kore e pērā, ka raru. Te mea ke he nui ngā huarahi hei a whina I a koe, ko te hia, ko te wawata o ngā tūpuna, kia noho tātou I roto I te aroha, I roto I te āio o te aohō. Mā te tautoko ka whai kaha, ka ora. Ko rai hapa ki tēnei e mihi atu nei.
Thank you, Raiha. So you can see that we try and make sure that the program is personalised to all of our students. One of the things that we know about adolescent females with severe dysmenorrhea is the huge impact, and this has been brought out already in the presentations today. Disrupted development, education, missed job opportunities, but this is the thing that I think that we're really missing, and that's the emotional, psychosocial, physical, and mental health. And it doesn't take too long when a young person has severe dysmenorrhea that may be endometriosis that she gets frightened of getting her period, and we have to stop that. Globally, we know that I'm getting feedback. Is that? I'll continue. Globally, the school absence due to dysmenorrhea is 20 to 31%. We also know that from the Australian study, it was the same 25%, in our study, 27%. So what we know is that early recognition of symptoms and timely intervention is absolutely crucial. And education really is the best screening tool we have at the moment. And so I urge you to think about this. We, of course, follow a multidisciplinary treatment. When we look at some of the studies that are around, we can see that this study is a very nice study looking at a huge cohort of 21,500 young women in a systematic review and meta-analysis. And they found lots of data, and in the interest of time, I won't go through it all, but the biggest thing here is that globally, what we're finding is 71% of our under 25s have severe or significant dysmenorrhea that interferes with their quality of life. So how do we get it out there? Well, we're just looking at that right now. So I've been spending the last year looking at how we might turn an in-schools program into an online program. And let's just take the New Zealand example, for instance. In New Zealand, we have 300,000 secondary school students. In 2018, the ME program going into each school reached 13,500 students. In its time, it's reached nearly 300,000 students. Now, when we did a survey out to our communities and schools, our community and our students, 98.5% said that they would use an online program. So when we think about e-learning, we think it's cost effective, it's efficient, it's environmentally friendly, and it's accessible. And it can improve well-being and health-seeking behaviours. So right at the moment, I'm doing pilots and tests in classes. And how do we get that through? Because we must have an equitable, medically responsible, inclusive education with no borders. And I think that's really important here. So the Me program is built in modular components, even though it's a nice flow through, so that it could be, we're trying to make it so it's a bit pick and mix. You can choose the modules that you might have. Endometriosis does not stop in a pandemic, and we need to think about this now in terms of COVID and the impact that this is having on the lives of our young people, how we get the message out there and how we ensure that we continue with our good work and COVID doesn't get in the way. So there are the survey comments. I won't go through them all in the interest of time, but you can see what teachers at school said, what students said, and post the trial, the students were saying, oh, I love the games because it does have gamification. They got a digital badge. They could do it during lockdown. And even one of our, a transgender young woman, she's only 16 years old, said how much she enjoyed it and thanked us very much for being inclusive. Here's another question I want to ask you. This is Laura. She's 15 years old with very mild stage endometriosis here and in the pouch of Douglas around in here. This is Lisa, who's 32, with severe stage 4 endometriosis, large endometrioma. I want to ask you, can we stop this from becoming this? And we don't know yet, but we can make a jolly good effort to try. And we think that certainly our early findings show that we are making headway. So I'm asking you to commit to stop the harm and the significant human and societal impact of dysmenorrhea and endometriosis. We have demonstrated that the In Schools Me program improves early diagnosis of endometriosis and management of dysmenorrhea in teenagers. So professional adolescent education is an appropriate screening tool. So let's collaborate. And I really took note of the introductory message that was given, I think, by Professor Chaudhry. Let's fight together to combat endometriosis. Me Online is currently under study at Otago University in New Zealand. We're looking at a nationwide rollout, and we're looking at also how this can have global relevance and implementation. Listen to the guidelines. Let's look at what they're saying, because we've just released the guidelines in New Zealand on the diagnosis and management of endometriosis. The RANSCOG guideline released this year. The NICE guideline in the diagnosis and management, 2017. The ESHRA guideline, which is currently up for review. The World Endometriosis Society consensus on the current management of endometriosis. All of these guidelines are saying early intervention, we must try and reach that goal. And I encourage the science to continue, but how do we also do that in very practical ways? In the National Action Plan for Endometriosis in Australia, they also, through a very successful trial in South Australia, supported the expansion of the Me program. And it's the only program endorsed by the World Endometriosis Society. I conclude with the following slides by now asking, from the similar take on the poem, how do we love and respect thee? And I think that that can be summed up beautifully with the World Health Organization tile, and I congratulate those of you using these tiles, by let's making sure that every person with endometriosis gets the diagnosis and care they need to manage their symptoms. It's a matter of health and well-being and human rights. I'd like to acknowledge and thank the students, the schools, and the teachers who have supported the Me program for 22 years. To my esteemed study team, Professor Neil Johnson, Ms. Emily Brick, and Mr. Michael East. To the Philanthropic Trust for funding the program over those 22 years. And to all of those who believed in the importance of menstrual health and endometriosis in schools through the Me program from very early beginnings. And a very big thank you to the EASB for their insightfulness and including this presentation in this scientific meeting. Thank you very, very much.
Thank you, Madam. Thank you for your excellent presentation.
Lastly, I'll just present that I hope that we can all share the science, the research, the practical management and treatment of endometriosis in Edinburgh in 2023. So we look forward to that. I thank you for this wonderful meeting. I thank you for asking me to present, and I hope you have found it a useful presentation.
Thank you, Madam. Thank you for your excellent presentation. Now, may I request our first chairperson, Professor Roshana Begum, to comment on the presentation of first speaker, Professor Khaled Khan. Professor Roshana Begum, Madam.
Thank you, Professor Diba, and thank you, our President Samina Chaudhry, Professor Saleha Chaudhry, and also Sharmina Abbasi. They are doing a hard job and a tireless job for this endometriosis, and I am grateful to them and for this session, and I am also grateful for making me a chairman of the session. First of all, you asked me about the Dr. Khaled Khan. Dr. Khaled Khan, and he is working in Japan, and he is very close to us. Thank you for your excellent presentation, for an excellent work. Actually, you are working in the Japan. Japan is a mainly research work is there. They are doing, and from this research work, you have shown an excellent presentation on the newborn baby, about the newborn bleeding, and study on the neonatal uterine bleeding, and how from this study, how can we prevent this endometriosis, and how we can actually do actually I was surprised. You can, if we diagnose them, we can prevent the ovarian malignancy also. We are also working on this session also, this study also. And with this study also, I have you asked me only to tell about the Dr. Khaled, and I invite my hats off to also our next speaker, Professor Debra Bush also, because I think that she was wonderful presentation on the human rights of the adolescents. Thank you. I am not going on details of everything. Next speaker, Professor Ferdowsi will cover that one. Thank you, Professor Khaled.
Thank you, Madam.
Thank you so much.
Madam.
Thank you.
Thank you, Madam, for your comment. Now, may I request Professor Ferdowsi Begum, Madam, our second chairperson, to comment on Professor Deborah Bush's presentation.
Thank you. Respected chairperson, chief guests, very distinguished speakers from all over the world, and also our colleagues, friends, teachers, students. I think this is a very good congregation of like-minded people, especially those that are interested to learn about endometriosis and help persons who are suffering from this silent but a cause of their suffering of women, especially the youngsters. I must be congratulating Dr. Deborah Bush from New Zealand, from Lanchcourt. I think you are from very, very far away country, a beautiful country. I thank you and your team from the core of my heart for the hard work you are doing. It's not the thing can be shown very easily in few slides, but the hard work behind it really counts. And the ME program, which you are undertaking, your group is undertaking, it's fantastic, which we learned about and lots to do about it as well, from learning from there. So of the 300,000 students, you reached about 13,000 students. So
in 1 year.
In 1 year. So it's a big way of doing things.
Yes.
And the way the adolescent reacted is fantastic. They really liked the program. So you converted the in-school program to online and very entertaining program as well. So we really thank you. And I think there are many groups in the world who are working in this in different way to reach the adolescent and make their life healthy. The question you thrown to all of us as a challenge, that how we can prevent those very tiny lesions of endometriosis to turn into stage 4 endometriosis. So there are many others who are working. And in this opportunity, I must be thanking Dr. Khaled Khan, whom I thank you so much, dear friend from Bangladesh, now Japanese. So
thank you so much. It's glad to see you after a long time.
But the thing is, even if you're Japanese, you cannot make your nose flat. So that's a failure for you. Sorry.
Thank you so much.
Yeah. So the work you have done is wonderful. It's called consistency. Starting from the bits, from the scratches to reach to the height of the mountain and still exploring. So that's very fantastic. And I think this work must be combining, you know, the way Professor Khaled Khan is working. I think that can be converted. This can be amalgamated, you know, throughout the world so that we can build on evidences from different angles. So that's very fantastic of these two people work. We are seeing one spoke about clinical aspect, the other told about the educational and social aspect. So this we welcome this kind of presentation, and I hope our youngsters and we will be learning that how to spend our time in research, in education, and also promoting this among our youngsters and reach them who are really suffering from this program. So this is always a fantastic session and adolescent. I must be congratulating the organizers and the fantastic presentation by our speakers.
Thank you, madam. We are already running out of time. May I request my co-host, Dr. Farzana Rinki, to wrap up the session.
It's my pleasure to thank our honorable chairpersons, the amazing speakers. It was really nice to hear. And thanks to Dr. Farzana Diba, my co-host. Now I welcome you all to the next session, the panel discussion. Thank you.
Respected ladies and gentlemen, respected ladies and gentlemen from home and abroad, good afternoon and Assalamu alaikum. I am Dr. Farzana Rashid. I would like to welcome you all on today's first International Virtual Conference of Endometriosis and Adenomyosis Society of Bangladesh. Today, our session will be on the panel discussion on the topic of challenges on preservation of fertility in endometriosis. Our session will be chaired by our renowned chairperson, Professor Nalini Kal Mahajan and Professor Musammat Rashida Begum, Madam. Both of the chairpersons are very renowned and stars in our fields. I have the honor to introduce them in front of you. Professor Nalini Kal Mahajan, she is the founder president of Fertility Preservation Society India, President Asian Society of Fertility Preservation and Chief Editor of Oncofertility Journal. Professor Musammat Rashida Begum, she is the Chief Consultant of Infertility Care and Research Center, Vice President OGSB and Board Member Aspire. Ladies and gentlemen, this session will be moderated by Professor Fauzia Hussain. She is the Professor of Department of Gynecological Oncology, Bangabandhu Sheikh Mujib Medical University. She is the International Affairs Secretary of OGSB, Clinical Lead for Protocol and Guidelines, RCOG, past Honorary Secretary of IRC, RCOG. So I would like to welcome our moderator, Professor Fauzia Hussain.
Professor Fauzia Hussain, unmute yourself, please.
Fauzia, madam, unmute yourself, please.
ফৌজি কি আছে?
Yes, only
Sorry,
absent.
madam.
Unmute
I was trying to unmute myself, but I couldn't because the host didn't allow me to do. Sorry about that. Distinguished guests, ladies and gentlemen, and thank you, Farzana Rashid. We all know that endometriosis is a common chronic inflammatory disorder, often causing both pain and infertility. It is estimated that 25 to 50% of patients undergoing fertility treatments had endometriosis, as it involves an impairment of the ovarian reserve. For these reasons, endometriosis has been highlighted as a condition that may require a fertility preservation while being benign in nature. Dear audience, in this session, we have a keynote paper followed by panel discussion. The speaker will have 12 minutes for presentation. If you have any questions, please send it in the chat box, which will be discussed at the end of the session if time permits. It is my pleasure to introduce our eminent speaker, Professor Angela Anguiller. She is professor and university scientist of the University of the Philippines College of Medicine, President, Philippine Society for Reproductive Medicine, past President, Pediatric and Adolescent Gynecology Society of the Philippines and Society for Gynecologic Endoscopy. She is the Scientific Committee Co-Chair, Asia Oceania Congress in Obstetrics and Gynecology, board member of Asian Society of Endometriosis and Adenomyosis. She received awards such as University Scientist 2020 from the University of the Philippines and the prestigious FOG Shanis Ratnam Young Gynecologist Award. She will talk on challenges of fertility preservation in endometriosis. Please welcome Professor Angela Anguiller. Thank you.
Thank you, Professor, for this kind introduction. I hope you can hear me and allow me to share my slides. So you can see it already, right?
Yes, ma'am, we can hear you and we can
Thank you. I shall proceed. So endometriosis is a challenging disease that have devastating consequences on a woman's fertility. Good day, I am Professor Angela Aguilar, and I will discuss how we can preserve fertility in a woman with endometriosis. And I have 3 points to make. The first is to discuss how endometriosis affects women's fertility, primarily her ovarian reserve. Number 2 is, what can we do to minimize the harm brought about by the disease? Number 3, and my last point, is to be aware of fertility preservation as an option for women with endometriosis. So let us first discuss the impact of the disease called endometriosis on fertility. In nearly 30,000 women surveyed in Canada, women diagnosed with endometriosis are 4 times likely to be infertile. Quite a big number, given that the prevalence is only 7% in that population study. That is because endometriosis affects fertility through various mechanisms. Apart from negatively affecting the pelvic environment through mechanisms such as anatomic distortion and immunological inflammation, disturbed folliculogenesis also occurs, and this leads to an impaired ovarian reserve compared with women of similar age and circumstance. Women with ovarian endometriomas have been documented to have decreased anti-Mullerian hormone levels. Their ovarian reserve can likewise decline faster over time compared with women without endometriosis. This was demonstrated in a retrospective study by Romansky, where patients diagnosed with endometriosis had a lower ovarian reserve compared with women with endometriosis. Look at the graph and compare the two curves. The lower bar represents women with endometriosis, and indeed their AMH is quite considerably and significantly lower. I'd like to show you data from our Center for Advanced Reproductive Medicine and Infertility at our St. Luke's Medical Center in the Philippines. This revealed that among those who underwent egg cryopreservation, they seem to exhibit a likewise steeper slope versus age in anti-Müllerian hormones compared with those without endometriosis. The pink ones are those with endometriosis and the green dots represent those without endometriosis. Although our numbers are small, the mean ages are similar in both groups. So they are comparable in terms of mean age. But if you look at the AMH, it appears lower at 2.17 in women with endometriosis compared to those without at 2.86. Thus, efforts should be made to minimize the negative impact of endometriosis on a woman's fertility. So how do we do that? Early clinical detection is key. Pay attention to the history, do minimally invasive diagnostics, and when you have reached the clinical diagnosis of endometriosis, as in this paper and recommendation by Agarwal, timely intervention should be done. What is appropriate intervention for women with endometriosis, especially for adolescents and young women? Long-term medical suppression is the mainstay of endometriosis because it can manage the symptoms, it can avoid surgery, and over a long term protects the ovarian reserve. In this study by Mutsi, it shows that by administering an effective progestin to suppress endometriosis, there is the regression of the cyst, the improvement in the antral follicle count of the affected ovary, and last but not least, there is maintenance of the anti-Müllerian hormone. And this is in contrast when you do surgery. In a paper by Eunice, cystectomy, especially bilateral, considerably decreases ovarian reserve. The effect is more pronounced in bilateral ovarian surgery, and thus surgery does not offer any advantage compared with medical treatment. In fact, recurrence is common, and worse, repeat surgeries are a reality, and this will aggravate what already is a negative impact of the first surgery. And perhaps the only room for surgery will be to enhance access to follicles for egg retrieval in assisted reproductive surgery or techniques. Avoiding surgery is therefore paramount. The effect is detrimental regardless of parameters used, be it anti-Müllerian hormone or antral follicle count. We are now aware that fertility is indeed threatened in women with endometriosis, and our management of her symptoms require us to diagnose the condition early, require us to suppress the disease effectively, and lastly, to minimize the harm, especially in performing inappropriate surgery. But should we be more aggressive than that? Shall we offer fertility preservation? Data on women undergoing egg freezing show us several advantages of this approach. While indeed it is viable technology, it is no longer experimental, same as with social egg freezing, there are concerns, including the cost, the invasiveness, and the other threats that may make it less suitable as a strategy to improve or preserve fertility in women with endometriosis. Allow me to show this algorithm, published by Llarena, and this suggests that women who may need to undergo fertility-threatening surgery may consider oocyte cryopreservation, most especially in those women who are of advanced age. Egg collection can even be done prior to the surgical extirpation of endometriotic lesions as well as deep infiltrating endometriotic nodules. Indeed, we have often done egg retrieval without doing surgery for endometriomas. Intact endometriomas in situ still allow us to collect those eggs. This practice is supported by an observational study which was conducted by Cobo, who determined the outcomes of women with endometriosis who had vitrified their eggs. Women with no surgery have a greater number of retrieved oocytes. Higher live births were seen in women or younger women who have had no surgery at all. For older women, the effect of surgery on their egg collection is equivocal. Thus, egg cryopreservation is useful and successful technology for women with this fertility-threatening condition. How much egg should we collect and freeze? A follow-up paper by the same group calculated the number needed to freeze for women with endometriosis to have a reasonable chance of a live birth. And this is interesting. 3 oocytes will yield a 5% success rate, but adding 2 additional eggs doubles this chance to about 11.2%. The curves diverge, as indicated in the pink box, after this threshold is crossed, and women freezing their eggs at a younger age stand a better chance of having a child, as you can see in the 2 curves. Surgery may not matter for women freezing their eggs when they are young, but for older women, surgery will decrease the number of eggs collected, and the cumulative live births described here as oocytes used means that women may need to undergo more cycles of egg collection had they undergone surgery, especially if they're older. And this would require more resources to achieve a similar outcome. Given that there are similar number of eggs, however, women above the age of 35 will likely have the same cumulative live birth regardless of previous surgery. The key here, thus, and the lesson is to obtain a similar number of eggs to achieve the same live births regardless of age. These are our comparative data on egg freezing without endometriosis and with endometriosis, as plotted according to age. You can see on the left that the purple dots are those number of eggs collected in women without endometriosis, and those purple dots on the right graph shows the eggs collected from women with endometriosis. While no conclusions can be statistically made, we at least have our own data regarding our outcomes for our patients. Our data is consistent with many reports that lesser eggs are collected in women with endometriosis. Here in our center, they average 8.88 versus 11.31 per collection among women without endometriosis. Further data collection will allow us to decrease the very wide confidence interval and allow us better estimates for our own population. ART thus equalizes the chances of women with endometriosis given that they have frozen eggs, and it allows them to achieve a pregnancy. And this is comparable with those undergoing IVF for other reasons. Again, this is our data from our center, which I presented in the Asia Pacific Initiative for Reproduction in Kuala Lumpur in 2015, showing that there is no significant difference in the chances of pregnancy, whether it be from endometriosis, women, or those without. What about ovarian tissue cryopreservation? When will it be suitable as a strategy for fertility preservation in women with endometriosis? Ovarian tissue cryopreservation is suitable for women who are unable to or unwilling to undergo ovarian stimulation. It will be appropriate as an opportunistic procedure for women who are undergoing surgery in the first place. Another consideration is that the total cost of the procedure from storage to reimplantation is far costly than just egg retrieval or freezing alone. So thus the technique of ovarian tissue harvest, which we commonly do in our center for oncofertility, should follow the same criteria. Here, the Edinburgh criteria is employed, but because this is used for oncofertility, the requirements are not the same, I would say, compared with women who have endometriosis. For OTC, endometriosis requirements for the technique and risk are not as clear as that for patients who are undergoing egg freezing for oncofertility. We have to, however, counsel appropriately without overestimating the benefit of fertility preservation in endometriosis, and we should not underestimate the risk the patient will undergo should we perform our fertility preservation procedures in women suffering from endometriosis. I conclude that we have to educate our patients on the negative impact of endometriosis on the ovarian reserve. We should be aware, both us and the patient, that we have to minimize harm by effectively treating the patient using medication while avoiding surgery. But, however, given this new technology and this new strategy, we should consider and counsel for fertility preservation all those women suffering from endometriosis. Consider oocyte cryopreservation early and plan for ovarian tissue cryopreservation if it is cost-effective and for women who may actually have to undergo surgery for an opportunistic harvest of ovarian tissue. With that, I conclude my talk. Thank you very much for your kind attention. And I would like to give thanks and congratulate the women and men behind the first virtual international scientific conference, Professor Samina Chowdhury, who leads this, the president of the EASB, and Professor Saleha Begum Chowdhury, the Secretary General of the EASB. Congratulations and thank you.
Thank you, ma'am, for your excellent presentation. Now I'm requesting chairperson of our session, Professor Nalini Kaur Mahajan, for her remarks on the keynote presentation. So over to Professor Nalini Kaur Mahajan.
First of all, thank you for inviting me to chair the session, and thank you, Professor Aguilar, for an absolutely excellent presentation. However, a few concerns that I would like to raise is that, going through all the literature that you have on the women, collecting oocytes between endometriosis and non-endometriosis patients, very rarely have, they included big endometriomas in that. And I feel if you have a big endometrioma, 8 to 10 centimeters in size, what would you think should be the way forward? Should you aspirate, coagulate the base, stimulate, and then go for some kind of definitive surgery? Or do you think that we should carry on with the stimulation? Because we know that with the stretch of the ovary and the majority of the ovarian tissue being taken up by the cyst, it is very difficult and for the oocytes to grow on that side. And this has been shown in studies that where on the side on which you have a big endometrioma, as compared to the opposite side where you don't have an endometrioma, you do not have the same number of oocytes. The second and very important point is that we say that we should do medical therapy for endometriotic cysts, and that subsides and therefore surgery is not required. But what is the length of time which is required for giving this medical treatment for the cyst to decline substantially? I have seen women who have been 1 and 2 years of, Dynagest, and there has been no significant reduction in the cyst. So should we have some kind of a cutoff period that if within this period of time the cyst has not decreased in size, we should move ahead and advise the young woman to go ahead with fertility preservation and then maybe subsequently surgery? Because another very important aspect is pain. Pain is the reason why actually most of the women get diagnosed. There are, of course, some women who are asymptomatic and you suddenly find an endometriotic cyst when doing an ultrasound. But majority of women find excruciable pain, and they do not, they want to be rid of that pain however they can. And these are some of the, thoughts and concerns that I have. And I would really love for you to address these 2 or 3 points that I have brought up.
Thank you, ma'am. Thank you, Professor Nalini Kaur Mahajan. Your queries have been noted and we shall discuss it in the panel discussion session. Thank you for your valuable remarks. May I now request our chairperson, Professor Mosammat Rashida Begum, madam, to say a few words on the keynotes paper.
Thank you, organizers, for inviting me to chair this session. And thank you very much, Professor Angela, for your wonderful presentation. Angela covered everything about the management of endometriosis challenges management and fertility preservation. Fertility preservation, actually, it includes both fertility sparing surgeries, that is fertility protecting surgeries, and tissue preservation. I think both includes in fertility preservation. So we have to focus on primarily how to manage the patients, particularly in young adolescents, girls, then unmarried girls, and married women who have no children. So we have to very alert about the steps of surgery, procedures of surgery, we have to follow strictly the fertility preserving surgeries. And first surgery is the best surgery, and which must be done by expert laparoscopist or surgeon. Secondly, tissue preservation. We can preserve oocytes, ovarian tissue, and embryos. One thing is omitted, I think. For married couple, it is best option to preserve embryos, because success rate is very high in case of embryo preservation than oocyte preservation. So if lady is married, then our first choice will be preserving the embryos. And what Nalini says, that if there is big endometrioma, either unilateral or bilateral, then it is very difficult to stimulate the patient to retrieve the eggs. So in that case, we can, if when we go for preservation, tissue preservation, when MS level is very low, and if the patient suffers from pain and we need to do surgery, then we will think about the preservation and embryo. So we have to go for aspiration, and result of aspiration and sclerotherapy is very, very good. So we can aspirate the cyst, then go for stimulation and collection and preserving embryos. And for unmarried girls, we can, in the same manner, we can stimulate and collect the egg, and we will preserve the oocyte. And for other cases during surgery, where there is no chances of giving stimulation, then during surgery, if patient badly needs surgery due to severe pain or big omas, then during surgery we can preserve ovarian follicles, ovarian cortex, and the result of ovarian cortex preservation is also good. I think that's all. Thank you.
Thank you, Madam. Professor Mosammat Rashida Begum, for your appraisal and explaining the step-by-step procedure we should follow for the fertility preservation in women with endometriosis. Now, ladies and gentlemen, we shall commence an exciting knowledge sharing session on endometriosis. Our respected panel members are Professor Rokia Begum. She is professor and ex-head of Department of Obstetrics and Gynecology, Chattogram Medical College Hospital, advisor of USTC Medical College. Professor Mariam Faruqi, she is infertility specialist at Labit Specialized Hospital and Hope Infertility Center. Professor Major General Lisa Chaudhary, she is professor and ex-head of the Department of Obstetrics and Gynecology, CMH Dhaka, and head of Fertility Center, CMH, and she is the cultural secretary of OGSP. Professor Jasmine Banu, she is professor and chairman, reproductive endocrinology and infertility department, BSMMU. She's also the director of institutional quality assurance cell of Bangabandhu Sheikh Mujib Medical University. Then we have Professor Mafruha Khanam Parak. She is professor and head of department of OBGYN, USTC Medical College, Chattogram, and infertility specialist and consultant of Surgiscope Fertility Center. And our last panelist is Professor Jannatul Firdaus, professor, Department of Gynecological Oncology, BSMMU. She's the organizing secretary of GOSB and joint secretary of FBSB. So my first question to Professor Jasmine Banu, considering the queries raised by Professor Nalini Mahajan Kaur and from the chat box, we have Miss Ruby, who is aged 16 years. She is suffering from progressive dysmenorrhea for 3 years, and ultrasonography revealed bilateral ovarian tumors resembling chocolate cyst. Size is 5 into 4 centimeter and 6 into 5 centimeter. Now, how to manage this girl considering her future fertility? So over to, my question is to Professor Jasmine Banu.
Thank you, Professor Fozia Hossain, for asking me a good question. Assalamu alaikum and good afternoon to all. My patient is adolescent symptomatic endometrioma. So my goal of treatment is suppression of the pain and suppression of the disease progression and considering the preservation of the future fertility. So I would like to manage this patient with maximize the use of the medical treatment and avoidance of the surgical treatment due to the fear of diminished ovarian reserve. Regarding the medical management, your first line of therapy is the Dynogest. 2 milligram daily for the continuous therapy, and it can be given.
Madam, unmute yourself.
Madam, please unmute yourself.
Okay. newer research, the aromatase inhibitor, cabergoline, simvastatin, and metformin are used in the newer drug for the adolescent endometriosis because it is the best, important, and time-demanding drug for the adolescent endometriosis because it suppresses the pain and reduces the size of the endometrioma without the suppression of the ovarian function. So we use the medical management for this patient.
Ma'am, I have a question. Traditionally, we use NSAIDs and OCP as the first line of treatment, but you mentioned about Dynagest. So what is the cause behind it? What is the evidence behind it, ma'am?
Previously, it was said that the OCP and the NSAID is the first line of treatment for the management of the adolescent endometriosis. But nowadays, upcoming the Dynagest is the better option than the NSAID and COC because the gold stain it shows that the 50 to 70% of the adolescent endometriosis not responds to the NSAID and COC. And Cochrane review did not find out any high quality evidence to support the relief of the pain in case of the NSAID and COC. So we not use in the first line of therapy in the COC and the NSAID. But sometimes we use the COC and NSAID as the first line of therapy when there is a patient is unable to bear the cost of the Dynagest. Then we use the COC. And also other cause we use that is the least side effect and it is the readily available.
Okay, thank you, ma'am, and we'll follow these instructions. This is a good take-home message for us. And what happens if the symptoms persist? Are you going to intervene surgically for Miss Ruby?
Thank you, Professor Fozia. And during the follow-up treatment, if the patient not responds to the medical treatment, then we counsel the patient for the surgical treatment. But before going to the surgical treatment, you need the fertility preservation. So we counsel the patient for the family member for the fertility preservation, that is the control ovarian hyperstimulation, oocyte retrieval, and the oocyte preservation is the good option for the fertility preservation. And if as there is a long gap between the management of the fertility status and the patient marriage, so there is a long gap, we can give the, after surgery, can give the suppression therapy for the long time, and due to the recurrent chance is the more 20 to 40%. And you can two option. One option is for the fertility preservation before the surgery, and another option is the post-surgical hormonal therapy for the long time if the patient is not married or there is a pain is persist.
Okay, thank you very much. Thank you very much, madam. So I have a question for Professor Rokia Begum. Madam, are you with us? So Mrs. Sumaya, aged 35 years, has been suffering from primary subfertility for 10 years. She suffers from severe dysmenorrhea, dyspareunia, and received GnRH analog for relieving pain. She is nulliparous, and her AMH is 0.9 nanogram per ml. She underwent laparoscopic cystectomy twice for chocolate cysts. Now again, she developed chocolate cyst. Size is 4 into 4 and 3 into 2 centimeter. And she also has unilateral hydrosalpinx. So how will you plan for management considering her low fertility status? Professor Rokia Begum, madam.
Hello?
Hey, yes, madam, we can hear you. Yes, madam, please.
Thank you, Faudia, my beloved student. You said the patient is 35 years, 10 years infertility, and AMA is 0.9, and he has a hydrosulfins.
Yes.
And what hydrosulfins?
And she also has bilateral.
She has a bilateral endometrioma. Size is
yes.
Size is 4 centimeter and 3 centimeter, and she has a hydrosulfins.
4.4 into 3.2 centimeter.
3.2 centimeter.
Yes.
And she has a hydrosulfin.
So how will you plan for management? Yes,
hello,
madam.
she has a hydrosulfin.
Yes, madam.
Yes, madam. Hydrosulfin. But
yes, madam, she has hydrosulfin.
For her management, if she has a hydrosulfins, and before proceed to ART, you must remove the hydrosulfings or has to be clip the end of the cornual end of the tube for disconnecting the tube to the uterus, because in presence of hydrosulfings, fertility rate of IVF is not good. It's reduce the chance of implantation. And next, what is about endometrioma? Whether I proceed to do the surgery or straight IVF. But because she had an hydrosulfings, I need to go to laparoscopy. And if we do the laparoscopy, but she had a bilateral endometrioma. Bilateral, but size is 4 centimeter and 3 centimeter, is not very big, but what her AMH is too low, and I have to see whether the antral follicle count is good, and what is the access of the ovary. And before proceed to surgery, before proceed for disconnecting the tube, I have to check the follicle, also the follicle number, because AMH is low. Follicle, it's suppose the follicle is less. Now what to do? If the ovary is not accessible, we can make the ovary accessible by mobilizing the ovary. And if the cyst, sometimes it is associated with a peritoneal cyst, or it is endometrioma, it has to be 4 centimeter endometrioma is not so big, but if it is not follicle, accessibility of the follicle is disturbed, then I have to be remove this one along during the time of surgery. During the time of surgery, this is my answer. In short, in summary, has a hydrosulfing, visible hydrosulfings by ultrasonography, and ovary is not accessible to the collection of surgery. And
thank you, madam.
Of the tube and accessibility of the ovary. And then if it is necessary to remove the cyst as far as cautiously by preserving the sufficient ovarian tissue.
Thank you, madam. Thank you, madam. Thank you. So my other question is to Professor Mariam Faruqi. Madam, Mrs. Raki, aged 25 years, has come to you with the complaints of primary subfertility for 3 years. She has severe dysmenorrhea and dyspareunia. For abdominally, a 20-week sized lump is felt. For vaginally, pouch of Douglas is full. Uterosacral ligaments are taut. Uterine mobility is restricted. Ultrasound shows a 10 into 10 centimeter size chocolate cyst in one ovary and 6 into 5 centimeter size cyst in the other ovary. Her AMH is 1.1 nanogram per ml. So what should be your plan of action for preserving future fertility potential? Madam, Professor Mariam Faruqi.
Yes, thank you, Dr. Fauzia, for your thoughtful questions. And Assalamu alaikum and good evening to everyone. We all know the same thing is this. We are discussing the same things, but we know that endometriosis is a chronic debilitating disease which is underdiagnosed, underreported, and under-researched. So overall, there is a risk of premature ovarian failure, and therefore we are thinking of fertility preservation technique, which is gradually being focused. In this girl, if we could diagnose her earlier, medical suppressive therapy could preserve her ovarian reserve. But now she has a big endometrioma, symptomatic, and she is again desiring pregnancy. So we have to go for cystectomy since the size is very big and symptomatic. And in this cystectomy, we have to gently handle with the ovarian tissue with of cauterization minimum to save the ovarian follicular loss. And considering her age, AMH level, oocyte, we have 3 options: oocyte, embryo freezing, or even ovarian tissue freezing we can consider here. But the first treatment is surgery, where cystectomy. During cystectomy, we have to ablate the endometrial endometrial tissue, extra ovarian endometrial tissue where there is, with minimum use of ablation. And we will encourage her for pregnancy. If not, every time it may not be possible, but she is seeking for baby. If not, then we can go for other medical suppressive therapy before starting for ART. But here we have to counsel regarding the risk for the reduction of ovarian function and even the loss of ovary for this patient. And we have to, and if she had undergone previous surgery, we have to be very much careful about losing her ovary this time. And cystectomy, if done prior to ART, is here will be beneficial for relieving her symptom and also for accessibility of the follicles. And we all know that the disease itself is causing harm, reducing fertility, as well as the surgical intervention and also the ablative procedure, which also further deteriorating her ovarian function. So we have to counsel the patient, her husband, and if possible with the parents. And very this endometriosis mass, there may be a recurrence rate of 50% after 5 years. And there is also rare, but there is chance of malignancy in case of severe endometriosis. And another thing is the risk of...
Madam, I have a question for you. Madam, I have a question for you. Mariam
Yes.
Farka, Madam, I have a question for
Okay.
you. I mean,
Yeah.
if you are thinking of doing ovarian cortical preservation, are you going to discuss it prior to the surgery?
Yes.
Are you going to counsel the
Yes. Yes.
prior to the operation?
Yes, counseling is most important because
Okay.
it is.
I have another question to Professor Jasmine because she uses, yes, madam. Thank you, madam. So I have a question to Professor Jasmine Banu. What is the role of PRP during ovarian cortical preservation? Professor Jasmine Banu.
Thank you, Professor Fozia Hussain. Okay, it is the role of PRP in the endometrioma following the surgery. That is for increasing the ovarian reserve. We can give the platelet-rich plasma for finishing the surgery and followed by the ART with the hope there is the more oocyte will get during the oocyte retrieval.
So it is the autologous PRP.
Autologous PRP should be given following the surgery in the cortical tissue.
Thank you, madam. So my next question is Professor Mafruha Parag. In case of infertile women who are in stage 1 and 2 disease of endometriosis, what is the protocol for controlled ovarian stimulation and intrauterine insemination? Professor Mafruha Parag.
Thank you. Thank you, Professor Fozia Hussain. And Assalamu alaikum, everyone. And special thanks to our speaker, Dr. Angela, for her excellent presentation. Actually, controlled ovarian stimulation and IUI were associated with good outcome in terms of live birth in women with minimal and mild endometriosis. Some study concluded that controlled ovarian stimulation and IUI may be considered an option instead of IVF, maybe or further surgical therapy in those women who had a surgical treatment for stage 1 and stage 2 endometriosis. In infertile women according to American Society of Reproductive Medicine, stage 1 and stage 2 endometriosis considered controlled ovarian stimulation with IUI post-operatively within 6 months. So we can go for ovarian stimulation now and with IUI. We know the induced ovulation to improve the pregnancy by boosting the ovulation or through the induction of multifollicular development. Option of ovarian stimulation, we all know that clomiphene, cited, letrozole, that means aromatase inhibitor, or gonadotrophin, urinary gonadotrophin, or RFSs, that means recombinant FSs. Sometimes recombinant LAs nowadays may be needed. And gonadotrophin, we can use FSAs or HMG started early in the menstrual cycle and monitor with the TVS, transvaginal sonogram, is the mandatory. Ideally, hormone assess also be done, and trigger of ovulation with the HCG when the follicular maturity achieved. So, significant improve of pregnancy rate with controlled ovarian stimulation with IUI in contrast to natural cycle IUI. So, we all know that FSH, we can start 3 to 5 days of the cycle, and it is increasing, and to monitor the follicle and estrogen 7 to 8 hours every 2 to 3 days initially close to a trigger, and to modify the dose of evidence if there is over response. So ideal is the 1 to 2 or 3 follicles, 18 millimeter or 22 millimeter to 22 millimeter size at the time of HCG administration. And also we look for the estrogen level below the 1200 picomillimole per liter. LH, to detect the spontaneous ovulation or LH surge, we have to prior the plan of HCG trigger. So in absence of spontaneous LH surge, we can induce the ovulation with the HCG. So IUI after 24 to 36 hours or sometimes 42 hours after HCG administration, we can go for the IUI. And we use 5,000 international unit of urinary HCG or the recombinant HCG, that means 250 microgram, we can go for IUI. So gonadotrophin stimulation with IUI leads to higher rate of pregnancy. And lastly, I want to say that IUI with controlled ovarian stimulation might be a good option achieving a pregnancy in this group of this woman with endometriosis stage 1 and stage 2 endometriosis. Thank you very much.
Thank you, madam, for this very elaborated discussion on controlled ovarian stimulation and IUI. So my next question is to Major General Professor Lisa Chaudhary. Madam, when should women with endometriosis and subfertility be referred for IVF or ICSI? When to start? And is ART in endometriosis, is it different? Madam Professor Lisa Chaudhary.
Assalamu alaikum. Can you hear me?
Yes, ma'am, you're audible. We can hear you.
Assalamu alaikum and very good afternoon to you all. Thank you, Professor Fauzia, for asking me very important question. And I also thankful to the organizer of EASB to give me the opportunity. First part of my question, that when we start for ART, as because the management of endometriosis related to infertility is challenging and always debatable. It is very difficult to give up our old ideas and as to delay the 6 to 9 months to make a pseudo pregnancy or pseudo menopause state. We should never to delay her fertility. We should fight for her pregnancy. Don't give the emphasis on minimal or mild endometriosis, as because my previous panelist told that in case of mild and minimal, we should go for controlled ovarian stimulation and IUI. And for endometriosis and infertility, we should not treat the patient like normal gynec. We should treat like a fertility specialist. There is a newer method, that is EFI, that is endometriosis fertility index, to grade the endometriosis and prognosis of pregnancy. If the EFI is higher, there is more chance of pregnancy. So in case of advanced endometriosis, ART, that is IVF ET, is the first line of treatment. There is no role of expectant treatment in case of infertility related to endometriosis. And in case of milder, that is earlier endometriosis, my previous panelist told that IUI, controlled ovarian stimulation, IUI for 4 to 6 cycles. If failed, we should go for ART. And the second portion of my question, that is what is the difference between ART in case of endometriosis? There are a variety of protocols advocated for ART in infertility, and the long protocol with prolonged downregulation appears to be preferred method of ovarian preferred method to have when there is good ovarian reserve, that is MH level is good, that AFC is good, then we can use this long protocol with prolonged downregulation to get the better M2 oocyte and better embryo. And prior GnRH analog 3.6 milligrams subcutaneously monthly 3 months we can suppress, or we can go for suppression treatment for 3 monthly 11.25 milligram. It is also advocated. When there is poor ovarian reserve and advanced age of the lady and there is low endometriosis fertility index, antagonist protocol may be used. If endometriosis is associated with adenomyosis, then LNG, that is levonorgestrel IUS, that is Mirena, should be inserted prior to IVF ET for better successful result. And endometriosis affects adversely in embryo transfer. So hydrosalpinx, like our Professor Rokia Madam told, it should be clipped or removed, and endometriosis should be ablated either by medical or surgical method prior to FET or ET. This is
Madam. Thank you for your elaborate explanation of the methods of ART in case of women with endometriosis. My next question is to Professor Jannatul Firdaus, a woman aged 32 years with endometriosis and subfertility, had a laparoscopic cystectomy, and there was incidental finding of endometrioid carcinoma. So is endometriosis a precancerous condition? Professor Jannatul Firdaus.
Thank you, Professor Fauzia. You bring up an important issue, and thanks to the organizer to include me in this beautiful webinar. Actually, several case control studies and cohort studies have been published so far regarding that address the potential link between endometriosis and ovarian cancer. The results of those studies have shown that up to 1.7% of patients with endometriosis, they may develop ovarian cancer later on, or malignant transformation may occur. And these transformations occur mainly in the ovaries than in the extra ovarian tissues. And the special epithelial ovarian tumor, special variety, that is endometrioid variety or clear cell variety, and an occasional mucinous variety may be there. And the cause of this malignant transformation is mainly due to continuous production of estradiol, prostaglandin E2, and also excess iron in the endometriotic cyst lead to oxidative stress.
Okay, my other question is, what are the clinical characteristics of endometriosis associated ovarian cancer?
Okay, thank you once again. And endometriosis associated ovarian cancer have certain clinical unique characteristics, like occur at relatively earlier age, 6 years earlier than the non-endometriosis associated ovarian cancer, and more likely to be in the early stage in low grade status, and also have good clinical outcome, low rate of recurrence. But one important thing is that 40% patient with this type of tumor, that is endometriosis associated ovarian tumor, may be associated with concomitant endometrial cancer. So any patient with endometriosis having abnormal uterine bleeding, endometrium should be evaluated before offering any type of treatment. Thank you.
So is it possible to predict the malignant transformation?
Actually, we face this type of scenario sometimes, and it is very difficult to predict because, in the sign symptoms of endometriosis mimic that of ovarian cancer, like bowel bladder symptom, heterogeneous fixed adnexal masses, having restricted mobility. But even though there are certain clinical characteristics that may give a suspicion that malignant transformation is happening, like increasing size of the endometrioma, bilateral endometrioma,
presence of ascites or presence of solid component or any evidence of metastasis.
Raised CA125.
And also raised CA125, the rate of change in the CA125.
Thank
and
you.
one thing I like to add, the laparoscopically, if we look at the endometriotic lesion, then it typically the appearance, that is powder burn appearance, it lacks in this type of malignant transformation. And we can confirm by doing histopathological examination by looking at the cellular atypia or architectural atypia.
Thank you.
Thank you.
Professor Janatul Firdaus. It has been a very interactive panel discussion session, and it has certainly helped us to formulate an evidence-based guideline in the future. So wrapping up, a systematic review of literature showed that embryo, oocyte, and ovarian tissue preservation are the preservation methods for endometriosis based on the risk of premature ovarian failure. In endometriosis, the fertility preservation techniques have significantly increased. However, further studies and research concerning the risks, benefits, and cost effectiveness are needed. It is clear from the panel discussion that women with endometriosis should be referred to ART specialists earlier. Newer methods of medical management with gynecologist has been recommended by Professor Jasmine Bano and of course from several studies. And there is a new drug, first oral GnRH antagonist called elagolix that is taken as a daily pill. It helps with moderate to severe pain for endometriosis, to relieve pain in endometriosis. Clinicians should not prescribe hormonal treatment for endometriosis pain control before surgery due to the chance of underdiagnosis. There should be a specialist endometriosis center with gynecologists who has expertise in advanced laparoscopic surgical skills, a colorectal surgeon and a urologist with an interest in endometriosis, specialist nurse in expertise with pelvic pain, then advanced diagnostic facilities for radiology and histopathology, and of course the fertility services. Evidence suggests that surgery before infertility treatment is beneficial in grade 3 and 4, and ASRM states that operative laparoscopy is better than simple diagnostic laparoscopy for spontaneous pregnancy in minimal, mild, or even stage 3 and 4 disease of endometrium. They recommend ovarian cystectomy instead of drainage or coagulation.
Nolini has got something to say.
Hey, madam, I'm just finishing mid-decline and operation for ovarian endometrioma and endometriosis, ASRM stage 3 and 4 in vitro fertilization as an effective alternative. And in women with minimal or mild endometriosis, IUI and controlled ovarian stimulation showed 5 times higher pregnancy rates. So it recommends GnRH agonists for 3 to 6 months before ART to improve fertility. And the short-term hormonal therapy for endometriosis-associated pain after surgery, it is not recommended because the outcome of surgery is not changed. So, madam, our chairperson, madam, Professor Nalini Mahajan, may I know your...
I'm sorry I raised the hand,
please.
But I would just like to say that all this demonstrates that there is still no consensus of opinion, despite the guidelines saying one thing. There are other, meta-analysis saying that diagnostic is better than doing a surgical procedure. So I think we have to individualize. What is very important to understand is what is the woman seeking? Is she seeking pain control? Is she seeking fertility? Is she seeking fertility preservation? And that is the way forward. That is how we have to go. And I feel for the big endometriomas, it would definitely be in order to aspirate a huge 8 to 10 centimeter endometrioma. Do not do a cystectomy. You may or may not do a fulguration. You do a, ovarian oocyte preservation, and then put her onto medical therapy that may actually help the cyst to regress faster. I have never seen an 8 to 10 centimeter cyst regress on any kind of medical therapy, be it GnRH agonist, Dynagist, anything at all. So I think to individualize as to what the patient is wanting before we decide on which way to go. And you will see that across the board, there is so much controversy on what you should do and what you should not do, that you really have to see what is the need of your own patient. And so far as the oral antagonist is concerned, it's going to take a long time before it reaches at least Asian countries, at least India. I don't know about Bangladesh, but it's not available.
Yes, madam. Yes, madam, you are very correct regarding this, because all women needs to be individualized, and we have to prioritize her needs to relieve her pain for fertility or for other things. So thank you, madam.
Thank you.
And
Okay, I'm sorry to interrupt.
Yes, sir.
I'm sorry to interrupt. Dr. Sharmin, all the foreign speakers are under your name.
Yes.
So please correct it. All the foreign speakers are
I'm
under
trying
your
to
name.
correct it, sir.
Dr. Sharmin
I'm
Abbas,
trying
so
to
please
correct
change it. Okay?
All right,
Yeah,
thank
I'm
you.
trying to
Thank you.
correct it.
All right. So, and lastly, I just want to make a comment that women with endometriosis history have significantly high risk of clear cell, low-grade serous, and endometrioid invasive ovarian carcinoma, according to a pooled analysis of case control studies, which was published in Lancet Oncology. So these were the recommendations from today's panel discussion. A warm and cherished afternoon to the most valued honorable guests from home and abroad. On behalf of Endometriosis and Adenomyosis Society of Bangladesh and the entire fraternity of the society, our sincere gratitude goes out to all the dignitaries and well-wishers for your support. Thank you very much for joining this session. Now over to Rapporteur Dr. Farzana Rashid.
Thank you so
Thank
much.
you so much, Professor Fauzia Hussain, for your excellent moderation. It's my honor to thank our honorable chairpersons, eminent speakers, and lastly, I want to thank all the dignified foreign speakers and all the speakers from home and abroad. And lastly, thanks to the panelists for their knowledgeable deliberation. So here I like to conclude this session and next to Dr. Utpala Mojumdar for the next session. Thank you so much.
And thank you, Dr. Farzana Sharmin, for being an apt rapporteur. Thank you.
Thank you, Professor Nalini.
Thank you.
Being with us.
Thank you, Nalini, and all the participants,
Professor
panelists.
Nalini.
Thank you,
Nalini. Professor Nalini Mahajan, Professor Rashida Begum, Madam, and the respected panel members, thank you very much.
Thank you.
Yeah.
So over to Dr. Utpala Majumdar.
Excellent discussion. Excellent discussion.
Ladies and gentlemen, participants from home and abroad, good day to you all. This is Dr. Utpala Majumdar, Associate Professor, Department of Obstetrics and Gynecology, Dhaka Medical College Hospital. Welcome you all to our first virtual international scientific conference of Endometriosis Adenomyosis Society of Bangladesh. This is the time for Symposium 2 session, and the theme is Targeted Medical Management of Endometriosis. We are honored to have Professor Salma Roof and Professor D.P. Borua as chairpersons. Our first chairperson, Professor Salma Roof. She was the professor and former head of Department of Obstetrics and Gynecology, Dhaka Medical College Hospital. She is the treasurer of OGSB and vice president, Fetal-Maternal Medicine Society of Bangladesh. Our second chairperson, Professor D.P. She is the professor of Obstetrics and Gynecology Department, Holy Family Red Crescent Medical College Hospital, and also the former International Affairs Secretary of OGSB. Dear audience, we have Dr. Naushaba Tarannum Mehta as moderator of this session. She is working as assistant professor at the Department of Obstetrics and Gynecology of Bardem General Hospital and Ibrahim Medical College. Now, I'm requesting Dr. Naushaba to conduct the session.
Thank you, Dr. Utpala Majumdar. Good afternoon and greetings from Bangladesh. Dear audience, we have three eminent speakers in this session. The speakers will have 15 minutes for their presentation, humbly requesting all the speakers to complete their presentation within time. We will give a soft reminder at the end of 2 minutes. If anyone have any question, please give it in the chat box, which will be discussed at the end of session if the time permits. It is my pleasure to introduce our first speaker, Professor Taisuke Mori. He's in the Department of Obstetrics and Kyoto Prefectural University of Medicine, Graduate School of Medical Science, Kyoto, Japan. Dr. Mori is rated as an advanced doctor by Medifind in the treatment of squamous cell skin carcinoma. His top areas of expertise in the field of gynecology are endometriosis and uterine fibroids. His clinical research consists of co-authoring 166 peer-reviewed articles in the past 15 years. Now, may we welcome Professor Taisuke Mori.
Okay, shall we get started? So, can you hear me?
Yes, we can hear you.
Okay. So, thank you for giving me a great opportunity to present our work today. So today I would like to talk about aromatase. Sorry. Okay. Oh, sorry. Today I would like to talk about aromatase as a target for treating endometriosis. So endometriosis, the symptom and endometriotic lesion are relieved after menopause, and the growth is promoted by estrogen. So it is well known that endometriosis is an estrogen dependent disease. Aromatase is the enzyme converting androgen into estrogen. Aromatase is expressed in a number of human tissues and cells. Interestingly, we and other group showed in endometriosis. Sorry, endometriosis here. Aromatase is highly elevated and causes a high level of estrogen. On the other hand, in normal endometrium, aromatase is not expressed. This scheme shows a positive feedback cycle in endometriosis region, whereas in red shows these stuff are upregulated in endometriosis tissues. Aromatase, estrogen, ER beta axis is crucial for the maintenance of the disease because ER beta is highly expressed in local endometriotic regions. And then estrogen is directly linked with the promotion in the inflammation here. It is known that various inflammatory cytokines such as TNF-alpha, IL-1 beta, VEGF, and COX-2 are upregulated. PTE2 produced by COX-2 expression activation induces aromatase, leading to local estrogen production here. This is vicious loop to develop endometriosis. Here, I would like to focus on the regulation of endometriosis aromatase activity here. We focus on aromatase activity. So what regulates locally a overexpressed aromatase in the endometriosis? Here, this is the data from Professor Sasano in Japan, microarray expression profile in breast cancer tissues to identify positive aromatase. A positive aromatase. Aromatase is here. Interestingly, ER-alpha, which is one of alpha nuclear receptors, is very close to aromatase. And then they evaluate the mRNA level in each tissue. In breast cancer tissue, there were significantly positive association of ER-alpha with aromatase here. While in the stromal cells, there was no correlation between them. So here, I would like to explain about PGC-alpha. PGC-alpha is one of the transcriptional factor beta interacting with nuclear receptors involved in various biological responses, For example, fatty acid import, angiogenesis, mitochondrial biogenesis, steroidogenesis. So various environmental stimuli, such as cold, diet, and hypoxia enhances PGC1 alpha expression. For example, PGC1 alpha interact with PPAR gamma, RXR heterodimers to stimulate fatty acid import and oxidation in skeletal muscles. On the other hand, PGC1 alpha cooperate with ERR alpha in uterine endometrial cancer to regulate angiogenesis. Thus, PGC1 alpha work tissue specifically. So far, however, so far in endometriosis, the significance and function of PGC1 alpha in endometriosis has not been unclear. So we first performed immunohistochemistry analysis here. We clarified PGC1 alpha positively detected with the nuclear cytoplasma of stromal cells in ovarian endometrium. As you can see, immunostaining intensity of PGC1 alpha in ovarian endometrium was significantly greater than in normal endometrium and utopic endometrium. We next performed immunohistochemistry analysis for aromatase. This is positive control with placenta, negative control with liver tissues. Aromatase was expressed only in ovarian endometrium. Interestingly, as you can see here, we observed consistent expression of PGC1 alpha with aromatase in ovarian endometrium. Our PCR also showed significantly positive correlation between PGC1 alpha and aromatase in ovarian endometrium. How does PGC1 affect aromatase activity in endometriosis? To clarify the mechanism, we used the primary culture endometrial stromal cells from each cell. So we performed the gain of functional experiment with transcription using these expression and reporter process. This slide shows a regulation system of aromatase gene. Aromatase gene has 9 translated exons here and multiple untranslated exon 1. Aromatase coding region is here, exon 2 to 10. As you know, aromatase mRNA expression is regulated by multiple tissue-specific promoters in untranslated exon 1. For example, here, where aromatase is known to be the most abundant, mainly use promoter 1.1. In benign adipose tissues, promoter 1.4 is used, while in breast cancer, promoter 1.3 to here is used. Like this, aromatase expression is controlled by tissue-specific promoters. So in endometriosis, it is reported that promoter 1.3 to promoter 2 is used. We performed exon 1 RT-PCR. As you can see here, promoter 1.3 is highly elevated in all ovarian endometrium, while not in normal endometrium or utopic endometrium. When the cells were transfected with PGC1 alpha, the promoter level in promoter 1.1, 1.3 to 2 were significantly upregulated. We next focus on this site, promoter 1.3 to 2. This site, aromatase promoter 1.3 to 2 includes nuclear receptor half site AGG TCS, right in the red, which several nuclear receptors like LRH1, ERR alpha, COOP, SF1 bind to. Reporter vector including this site here. Also, we inserted here the mutation here, like this. So we designed the Lucifer's promoter reporter vector, including this site. So our result here, PGC1 alpha enhanced luciferase activity of promoter 1.322 in ovarian endometrium stromal cells. We next performed chip assay to evaluate the interaction of PGC1 alpha with nuclear receptor binding site, ERRE. So interestingly, PGC1 alpha was recruited to the genomic sequences containing nuclear receptor half site in ovarian endometrial stromal cells. We next investigate how PGC1 alpha affect the expression of aromatase. Our real-time PCR showed PGC1 alpha significantly increased the expression of aromatase and SF1, while such response was not detected in normal endometrium and utopic endometrial stromal cells. The same results were found aromatase enzyme activity here. Then, what regulates PGC1 alpha in endometriosis? So, endometriosis is regulated to many factors. For example, estrogen, cytokines, TNF alpha, or environmental stimuli, hypoxia exercise fasting here. So in our study, hypoxia and estradiol did not affect PGC1 alpha expression at mRNA level. Our results showed that TNF alpha stimulate PGC1 alpha transcription in ovarian endometrium stromal cell. Additionally, so we showed PGC1 alpha induced aromatase expression and enzyme activity in ovarian endometrioma, but not in normal endometrium. We also showed TNF alpha elevated in endometrial tissues, in turn induced the expression of PGC1 alpha. These findings suggest that PGC1 alpha could be a promising target for the endometriosis. So thus, we found PGC1 alpha contributes to enhance local estrogen production through aromatase. Then our next question is how PGC1 alpha play a role in the development of endometriosis. So we aimed to clarify the effect of PGC alpha on the cell proliferation and local inflammation. We performed proliferation assay. Interestingly, PGC alpha here promoted the cell growth in endometrium. Next, so we performed a loss of functional experiment with siRNA for PGC alpha. As you can see, the inhibitory effect of PGC alpha siRNA was confirmed by real-time PCR. So PGC1 knockdown significantly reduced the cell proliferation in ovarian endometrium. However, we don't know any ligand for PGC1 alpha. So as shown before, our chip assay reveals that PGC1 alpha recruited the region containing nuclear receptor half site here. Therefore, to screen ligand for the PGC1 alpha, we established luciferase reporter assay system containing this site. We tried many compounds of these here. HX531, which is RXR alpha antagonist, possessed the inhibitory effect for PGC1 alpha in ovarian endometrial stromal cells. So we next examine the prophylactic effect of HX531, this compound in OE and NE. As you can see, this compound significantly suppressed PGC1 alpha induced cell proliferation of ovarian endometrial stromal cells in concentration-dependent manner. We also examined the effect of this compound on aromatase expression. This compound significantly suppressed PGC1 alpha stimulated aromatase expression. Also, our exon 1 specific RT-PCR shows the same effect. So we examine the effect of PGC1 alpha on inflammatory cytokines. So we also Our real-time PCR also PGH1 induced IL-6 and IL-8 expression level in ovarian endometrium.
Sorry to interrupt. You
In
have
summary, PGH1 plays an important role not only in local estrogen production, but also in local inflammation, cell proliferation. Interestingly, HX531 suppressed PGH1 induced cell proliferation and expression, including aromatase, IL-6, IL-8. Thus, PGH1 could play critical roles in the development of endometriosis and be a novel target for the treatment of endometriosis in the near future. Thank you for attention.
Thank you, sir. Thank you, sir. Thank you for sharing with us your experience regarding the aromatase as a target for treating endometriosis. Now, may I invite Professor Juhi Agarwal. She is the professor at the Department of Obstetrics and Gynecology, Gandhi Medical College, Bhopal, Secretary of Bhopal Obstetrics and Gynecology Society for consecutive 2 years. Her presentations has been awarded in several times, including RCOG, West Zone, and West Zone UVA Foxy, and many more. She will be giving a talk on role of Dynogest in endometriosis. The floor is yours, Professor Juhi Agarwal.
Thank you, Doctor, for your kind introduction. First of all, I would like to congratulate the EASB Society for organizing this lovely virtual conference, which is very rare to see, and it has been, the topics are very good. And I will also thank the organizing committee, Professor Sahla Khan, Professor Samina Chaudhary, Dr. Firdausi, and Dr. Saleha Begum Chaudhary for inviting me as faculty. And I will not forget Dr. Sharmeen Abbasi, who have been in constant touch with me and working hard since last 1 month for the conference. So I would like to share my screen. Please allow me. Sorry, just have to adjust it a bit. Yeah. Is it visible, madam?
Yes, it is visible. You just have to give the slideshow.
Yeah. Yeah. Now is it visible?
Yes, madam, it is visible.
Yeah,
Thank you.
Okay. So I'm just a brief overview on endometriosis. We are all aware, I need not go in detail, that it's a chronic, which has to be emphasized, it's a chronic estrogen-dependent disease. It's presence of endometriotic tissues, ectopic as well as inside the uterus, causing inflammation, pain, resulting scar and multiple adhesions. And most importantly, it is affecting 2 to 10% of the general population, and 50% of the women who are infertile suffer from endometriosis. The incidence of disease is more, that means 1 in 10 women, which is a larger chunk. So prevalence of endometriosis, as you are aware, in India alone, I could have imagined all over the world, even in India, it is 176 million women who are suffering from endometriosis globally. And in India, it is 26 million. So it usually starts affecting from pre-adolescent age group. We are aware now, it has been proven, and continue till reproductive age group. And 20 to 29, that is 46% of the women, 46% of the women who are too young and are more productive in their years, they suffer from endometriosis. So these are the various sites. I will not go in detail. We are aware about the endometriotic lesions, which we see on laparoscopy. These are the powder burn appearances. We are worried about the ovarian endometriosis. Yes, the endometriomas, chocolate cyst, large endometriomas can occur, and the panel description we have, it was emphasized that it may also harbor malignancy. It is a marker of extensive disease. Now, deep infiltrating endometriosis is worrisome and increases the morbidity, which is higher grades like grade 3 and 4. 3 Ds are very important, that is diffuse chronic pelvic pain, dysmenorrhea, dyspareunia, and other like dysuria and dyschezia, which are distressing for the women. Causes of endometriosis associated pain are growth factors and the inflammatory markers, as as previous speakers spoken of. Bleeding from endometriotic implants, nerve irritation by endometriotic implants. Now, this is very important slide because it causes delay in diagnosis. So, start of symptoms may occur a year before, first consultation may occur after an year, and the diagnosis may take as long as 7 years, which is a long time. Till then, the damage has already occurred. Now, the risk factors, I will not go in detail. We are all aware of. Now, this is very important thing. As far as impacts of women and society is concerned, it affects the sexual life, the relationships and the family. The young girls, their performance get hit at the level of school, university and work, physically and emotionally debilitating and significantly reduces quality of life. So the quality of life is, as the pandemic was, we have all faced pandemic and we know that quality of life is the most important thing which everyone aspires for. So endometriosis is a chronic disease which can affect the quality of life significantly. The goals of management. So given the chronic nature of the condition, the modality should be always balanced in terms of long-term acceptability and also the safety profile. And we need symptomatic treatment, avoid delay in diagnosis and reduce unnecessary surgery, of course. Now, what type of molecule may be good for endometriosis? A molecule which can be given for long term, may have minimal side effect, effective for pain. Yes, pain is a significantly involved in endometriotic lesions. Not to deplete estrogen completely, because we do not want side effects of estrogen depletion like decreased bone mineral density. Act on implants and endometriomas and may cause minimal recurrence. There are various medical management options to like first line management, maybe combined hormonal contraceptive pills, progesterone only. Second line maybe GnRH agonist, LNG IUS. We have been discussing those constantly. My previous speaker spoke about aromatase inhibitor and NSAID. But the first line, we'll talk about first line, that is progestin. Now, how do progestin act? They act by reduction of serum estrogen level, which is the chief culprit in causing endometriosis. It causes immunomodulatory effect. It has anti-inflammatory effect. It causes deserialization of the endometriotic implants. It inhibits metalloproteinase and also has anti-angiogenic effect. So these are all desirable effects in endometriosis as far as the clinical implications of endometriosis are concerned. Now, what is Dynogest? Why we are talking about Dynogest? It is a progesterone. So again, it's a 19-not-testosterone derivative with a cyanomethyl instead of ethinyl group at C17 position. And it makes it is a compound with fewer hepatic side effects. That means it is safe to give in long term. It comes by various names like Dynogest, Vizana, Salkia. There are various trade names available. Now, what makes it as an ideal choice for endometriosis? First of all, the boxes, you can see that it has a strong progesterogenic effect. It has good tolerability. It has least anti-androgenic effects because certain progesterones have pronounced androgenic effect. Relatively moderate inhibition of gonadotropin secretions, which handles its long-term use and reduce bone mineral density. High oral bioavailability and also has a peripheral action. Now, here is a comparison showing Dynogest with other progesterone. Its progesterogenic activity and least anti-androgenic activity makes it an ideal molecule for its effect on endometrial implants and also for its long-term use. Now, how does actually Dynogest act? It has both central effect as well as peripheral effect. So central effect, it inhibits gonadotropin secretion and hence it's hypoestrogenic. And also it causes anovulation, again causing further estrogen depletion. Now the local effects are, it is anti-proliferative, anti-inflammatory as well as anti-angiogenic. Now coming to pharmacokinetics, since we have to give this drug for long-term use and it has to be, the action has to be rapid. So it is rapidly and completely absorbed after oral administration. It peaks in 1.5 hours, 91% bioavailability, 9 to 10 hours of half-life, not related to food, does not bind to serum hormone binding globulin. And so this is how this pharmacokinetic help us for its long-term use, its fast action, low dose can be given, and it has no side effects, and it does not get affected by any of the drugs given if any of the drugs are given simultaneously. So ASHRAE and West consensus is progestogens are the first line and Dynogest is the. So in order to prove whether Dynogest is the ideal preparation and it should have all the qualities, that means it can be used for long term. It has no side effects of estrogen depletion like depleting bone mineral density. It should improve quality of life. It should have comparative subsidence in the endometriotic lesion as compared to GnRH analogs, and its comparison to laparoscopic surgery, and also its safety profile as far as our adolescent population is concerned. So various research trials have been conducted. Now I will come into each one by one. So first, this was a research trial which was done for efficacy and safety of Dynogest for 65 weeks. So it's long-term use. So here what was done was, here there were two groups which were divided, and out of these two groups, some of them were given Dynogest and other were given placebo. A significant effect was seen in the pain threshold and the frequency of intake of analgesics. So that proved that there was a significant reduction in pain when it is given for long-term use. Now here you can see the bar score. Now there is significant fall in bar score in the Dynogest group as compared to placebo. Though there were certain adverse events as well, like breast discomfort, nausea, irritability, but these side effects were very moderate and mild, and low rate of discontinuation was observed with these women after long-term use. There was significant reduction in pelvic pain and reduction in endometriotic lesions, though it was associated with certain bleeding irregularities. Now coming next to dose ranging. Now, which dose is adequate as far as Dynogest was concerned? So this study was done in 2010 to see for the dosage. So various doses were tried for, that means 1 milligram per day, 2 milligram per day, and 4 milligram per day. And a second look laparoscopy was done after giving these various doses. Now, it was observed that endometriosis related symptoms like dyspareunia, dysmenorrhea, diffuse pelvic pain, and premenstrual pain were subsided. Adverse events were mostly mild to moderate in intensity, and there was no notable side effects of lipid, carbohydrate, and liver metabolism. So the results were similar to the 2 milligram Dynogest was accepted, and the results were also then compared to leupride acetate in another study, and it was seen that the results were comparable. Now here is a study which shows the DNG versus leupride acetate. Now here you can see that both the results are parallel to each other. That means the pain related factor, the reduction in pain was similar in both the groups, whether it is leupride or whether it was DNG. So it was proven that it is as efficacious as LA. Coming to bone mineral density, it was observed that there was significant fall in bone mineral density as far as the LA group was concerned. And also the hot flashes, which is very distressing for the woman. And as far as the young girl is concerned, the hot flashes are very distressing. So you can see that that was more in LA group than Dynogest. Now, we concluded from these studies that yes, Dynogest is a safer and better molecule, and also a molecule which does not affect quality of life. So coming to endometriosis related pain, no women had severe pain, only 5% had moderate symptom, no clinically relevant impact on standard hematology. There was a favorable safety profile, lower incidence of hypoestrogenic effect, and limited bone mineral density changes as compared to GNRH. On recurrence of ovarian endometriomas more than 2 centimeters and severity of pain after laparoscopic surgery. You can see that the p values are significant. So this suggests that DNG also reduces the lesions on laparoscopy. Now in DNG group, there was no recurrence, but in expectant group, there was significant recurrence. You can see this. Now here they are talking about the post-operative recurrence. Now here also the post-operative recurrence in DNG versus no medicine group, you can see that it was a retrospective cohort study for 5 years. Ovarian endometrioma excision was done and TVS was done every 6 months to see the size. In DNG group, there was no recurrence. I mean, that is not significant, only 4%. But in no medication group, it was 69%, which is quite significant. Coming to infertility, we have been talking infertility since morning. So a meta-analysis have shown that chance of achieving pregnancy after ART was significantly lower in patients of endometriosis because it reduces ovarian reserves. So it is reasonable to assume that if the probability of pregnancy after ART significantly decreased, then treatment of endometriosis immediately before ART cycle might be associated with improved outcome. So here you can see the ovulation inhibiting effects of Dynagest, which we are aware of. But what is more important is subsequent suppression endometrial growth in the lowest dose, that is 2 mg. This result indicates a potent endometrial effect on Dynagest. How does Dynagest act on the follicles? There is reliable ovulation inhibition while maintaining LH and FSH ratio, which is very much important to maintain the bone mineral density and a threshold estrogen level during rest of the cycle. Apoptosis of granulosa cells occur of dominant follicle, leading to decrease in estradiol level while maintaining the FSH level, which is very important. There is strong peripheral ovarian effect inhibiting the estradiol production. So the return of ovulation after Dynagest, an LH surge in urine was identified during follow-up between day 1 and 43 after cessation of treatment, and progesterone concentration was above 1.6. That means the ovulation returns fast after about 2 months after giving Dynagest. So once you suppress the endometrial lesion after Dynagest, the ovulatory function resumes after 2. There is also return of regular menstruation approximately after 1 month of giving Dynagest. So here is a clinical evidence showing that outcome of Dynagest treatment followed by in vitro fertilization and embryo transfer in infertile women with endometriosis. What they concluded was that if you try IVF in ET immediately after Dynagest treatment, you may not be successful. So you need to give some time so that her ovulatory function resumes, and then you can go for a treatment. So here is another which is concluding that Dynagest to be possibly more efficient treatment option for these patients who have ovarian endometrium. Now we are all aware about this huge study which has been carried out in 31 European countries for adolescent women, where the safety of endometriosis, safety of Dynagest was already established in adults, but was still doubtful in adolescent people. So this was an open label enrollment done in these countries. So all the girls were subjected to this 2 mg Dynagest per day for 52 weeks, and bone mineral density was seen at the end of the treatment. And also a 6-month follow-up was done. So no significant decrease in bone mineral density was found. And also there was significant reduction in pelvic pain even in the adolescent girl. You can see the pelvic pain reduced significantly, dysmenorrhea also, and dyspareunia. Now here you can see patient satisfaction. This is very much important as far as treatment protocols are concerned. Though it may be scientifically proven that yes, it may suppress the endometriotic lesion, but still if the patient satisfaction score is poor, the drug is of no use. So here you can see that Dynogest has proven to be a very good drug as far as patient satisfaction is concerned. Therefore, there were approximately 84% of the women who were very well satisfied after giving Dynogest. So conclusion, we have already discussed. I will not go in detail. This is comparative of all the studies which I have quoted. You can see that there was a significant reduction in endometriotic lesion, pain, quality of life, acceptance. Now coming to dosage. So how to prescribe Dynogest? 1 tablet daily for minimum of 3 months to maximum of 5 years continuously can be given. It can be taken at any day of menstrual cycle. Can be taken with or without foods. Now there are certain other application of Dynogest as well. It is an appropriate choice for post-premenopausal women who have need of contraception. So it can be given with estradiol combination as a contraceptive method, and to the women who are not desiring to get LNG IUD inserted. It can be given in women with heavy menstrual period, in sexually active women with anovulatory or ovulatory cycle. It can be given in heavy menstrual bleeding in non-sexually active women, heavy menstrual bleeding who plan to conceive in an upcoming year, and also in heavy menstruating women with concomitant condition, that means endometriosis. There are certain contraindications, of course, as it's a pharmacological agent, it has to have contraindications. So you have to be very aware if the woman is having a previous episode of venous thromboembolism or an arterial thromboembolism, you will never prescribe Dynogest. CVA, history of CVA, again, migraine, undiagnosed vaginal bleeding, undiagnosed amenorrhea, and also hypersensitivity to any of active substance or any other excipients. So you have to be very careful in these women who have these problems. There is a certain problem, that means certain bleeding patterns are there in case of Dynogest. Initially, there can be desynchronized bleeding patterns. So you need to counsel the patient. Counseling is very important. We have been talking about counseling since morning, that in this disease, since the nature of disease is chronic, it's very important to counsel that you have to take this drug chronically. There may be initial some decentralized bleeding pattern. Later, there may be progressive reduction in bleeding days followed by amenorrhea, because many times women do not accept amenorrhea, especially the young women. So you have to tailor and you have to explain her the need of amenorrhea and about her disease, and also chronicity of her disease. Considering adverse effect, as we have talked about in our various studies, there are few side effects like headache, breast discomfort, depressed mood, and acne. But it is in very few women. But you need to counsel regarding these as well. Return of fertility, we have already talked of. Within 4 to 6 weeks, ovulation resumes. And from 29 days to 2 months, it may resume in 90. There are various studies. But still, it is not a licensed drug for contraceptives. So you have to be, in India at least, it is not licensed as contraceptive. So we don't use it. Now here is a lovely chart showing the threshold level of estrogen. And this is how Dynogest acts. And this is the threshold level which helps Dynogest to regain its popularity and also acceptability in young women of our endometriotic women. As far as DNG compliance is considered, it is treatment compliance during the long-term study was as high as 98%. Discontinuation rate, now these conclusions are from International Journal of Women's Health. So these are authentic, and yes, we have already seen it in our clinical practice. Discontinuation rate due to adverse events or lack of efficacy were both very low. Patient satisfaction with Dynogest, 88.9%, which is significantly high, and most of the women will be certainly willing and would prefer to use Dynogest again. So the take-home messages are, DNG 2 mg is an effective and generally well-tolerated treatment option. It reduces pain, hence it improves quality of life, reduces lesion, hence it is also useful in treatment of infertility. It improves quality of life, has an acceptable side effect profile, suitable for long-term use. Thank you.
Thank you very much, madam, for your evidence-based presentation. Now, may I request our third speaker, Professor Sanjay Kalkar. He is the clinical lead ONG, Basildon Hospital. He is also the consultant gynecologist, Southern University Hospital and NHS Foundation Trust. His special interests are endometriosis, minimal access surgery, including laparoscopic hysterectomy, and many more. He will be giving a talk on oral GnRH antagonist, that is elagolix, for the management of endometriosis. Over to you, sir. Professor Sanjay Kalkar.
Hello, assalamu alaikum, everybody. Thank you, everyone, for attending this wonderful conference. I am heartened by the enthusiasm shown by you all. And I'm just going to share my screen and we'll start in a minute.
Walaikum assalam.
Okay. Can you all see my screen? Yeah.
Yes, sir.
Okay.
Thank you.
Okay. Okay, so I'm going to, my topic is about oral GnRH analog, antagonist, sorry, elagolix for the management of endometriosis. I'm going to keep myself to the topic which has been given to me. So I'm going to talk about elagolix history and pharmacokinetics. I'll talk about the evidence. And we'll also talk about the clinical evidence, and I'll conclude what my take on this one is. Now, elagolix is a oral gonadotropin releasing hormone antagonist. It has been licensed for the endometriosis pain. And it is also trial for fibroid and heavy periods. And you might have seen that recently they published in New England Journal that it is effective in fibroid and heavy menstrual periods as well, along with the oral estrogen. So it's first developed in 2008 and licensed for medical use in 2018. I must say that it is not available widely, and I was actually, in fact, in UK, it is still not used, as in it has not been introduced in European Union, maybe because of the cost. But I was actually very pleased to see that in Bangladesh, probably you've got it available. So maybe we can import from Bangladesh, really. So it's only a few countries like US, Canada that it's being used. So basically, it's a small molecule non-peptide GnRH antagonist. So therefore, it can be administered orally. Whereas the peptide GnRH antagonist will be there will be a protein adhesiolysis, and therefore they cannot be used orally. And this is very welcome addition because it can be used orally, and some people would probably prefer, and you can always stop it at any time. Now, basically, how does it act? It acts by rapid dose-dependent suppression of pituitary gonadotropin, mainly LH and lesser extent FSH. So ultimately, all the efficacy of all the treatment we have been talking about so far is due to a hypoestrogenic state. So in a way, it's sad that it is still acting by the same mechanism. Whatever we've got other methods which we had so far is all by hypoestrogenism, and elagolix is not actually different. It is also acts by hypoestrogenism. And it starts straight away, peak effects 4 to 6 hours after oral administration. Now the dosage, you can tailor the treatment. Either you can give 150 milligram once daily. This is recommended for up to 24 months, whereas if you're giving a higher dosage, which is 200 milligram BD, it's recommended for 6 months. What is the evidence? Is there any evidence? So the biggest research on this published in New England Journal by Taylor et al. In 2017. But we must remember that this is a trial which is sponsored by the drug manufacturer. So there will be a downside to any trial which is actually sponsored by the drug manufacturer. So it's basically a randomized controlled trial of 1,689 premenopausal adult women. So basically there was 2 stages to it, 1 and 2. So basically all the women who were actually recruited into the trial did have endometriosis or had treatment with laparoscopy or laparotomy in the last 10 years. So there was always an evidence that it was endometriosis was present. It did not include women with a history of non-response to GnRH agonist or antagonist, cefomedoxy, Depo-Provera or aromatase inhibitors. In a way, because it is important that in these trials you have a pure cohort, but however, it is actually disheartening that it did not include those women who we find as clinicians difficult to treat. So what was the results? So basically, this is the phase 1 trial, which half of the patients were there. So you can see that compared to placebo, when Elagolix 150 was taken, there is a 46% reduction in dysmenorrhea, which is a fantastic news. And if the dose was higher, then it like 200 milligram twice daily, the decrease was actually much more pronounced, and it was sustained at month 3. There was benefit, and you can see that at month 6 as well, there is a similar sustained improvement, whereas with the non-menstrual pelvic pain, as the months when they're using, the benefit improves. So it's certainly statistically significant compared to a placebo. That means it does work, and the dosage is probably higher the dosage, the better response, but you have to titrate with the side effects of it. Similarly, the same results were obtained in the LRS trial 2, where first one is a half and it was a combined results, which I was talking about. Now, what about the other results? So again, in this chart, I showed that the results, the 3 major reasons why women may consult us, like dysmenorrhea, pelvic pain, and dyspareunia. There was improvement in all aspects in this trial. So the pain reduction was like 80% reduction, month 12, 78% with the 200 milligram dosage. So the same response has been found across dysmenorrhea, non-menstrual pelvic pain, as well as dyspareunia. So it is showing that the fact that it was published in New England Journal, and there is a definite sort of improvement in the symptoms. Therefore, the FDA approved it in 2018 for usage in US. This study concluded that both higher and lower doses of Elagolix were effective in improving dysmenorrhea and non-menstrual pelvic pain during the 6-month period with endometriosis associated pain. Now, how do we decide who are the patients we're going to treat with Elagolix? So basically, we're going to the candidate patients could be premenopausal women who are surgically or clinically diagnosed with endometriosis. So I was talking about the trial, which included only laparoscopy proven endometriosis. However, we know that as clinicians, when we examine a patient, classical symptoms, pain on exam, bimanual palpation, heavy periods. If you're suspecting anyone with clinically endometriosis or some ultrasound evidence of endometrioma, small endometriomas, we can always clinically diagnose. We don't have to do a laparoscopy in these women because laparoscopy is not always indicated. Again, it's got a cost, laparoscopy, and it's got its own risks, because we all know that in spite of our best intention, anytime you touch a patient, there is a risk. And can we reduce this harm by clinically diagnosing? And if those women who want a medical treatment, can we treat with them with just a medical management? I know as surgeons, we all like to operate. That doesn't mean that more and more medicine is moving towards non-intervention. So ultimately, as we all have said before, it's the women's choice, and we have to empower the patients we treat with the information, and they make their decision. So the indication could be those who have got endometriosis associated pain, and we have excluded other causes of the pain. Then if there is an endometriosis associated pain, we could try that. If they are somebody who have not responded to traditional treatment, i.e. you could just have your non-steroidal anti-inflammatory drugs, combined pill, or the progestins, we could potentially consider as candidate for this treatment. Or they could be somebody who's progesterone resistant disease, because you've given them a course of either progesterone only pill or norethistron, and it hasn't worked really. And then these are the candidates where you may want to try them. And also, if somebody had side effects with oral contraceptive in the past, like weight gain or any other concerns they may have had, and there may not be lots of women who we cannot use oral contraceptive pill, maybe smokers, high BMI, lots of contraindication. In these women, we can consider elagolase. Okay, do we need to be cautious in some patients? Yes, we shouldn't be using it probably if in the past they did not respond to GnRH agonist or antagonist, then it may not really work in this scenario. And we shouldn't be using with the CYP3A inhibitors. And also, we need to make sure that we are monitoring when we are using it together with digoxin and rifampicin. And dose increases with midazolam and rosuvastatin, and therefore we just need to be careful with them. What are the side effects? So basically, I said in the beginning of my talk that the side effect, it's actually a hypoestrogenic state. So basically, any symptoms a menopausal woman would actually face the same side effects, like hot flashes, night sweats, headache, nausea, and they can't sleep, brain fog, and obviously they may not have a period. And lots of women, this is a welcome side effect, but whereas I know some women, for culturally, probably, I don't know, in our cohort of patients, particularly culturally, Asian background women prefer to have the period. So for these ones, it may not be acceptable that they are not having the periods, whereas I see my white Caucasian population are always delighted not to have periods. So there could be a cultural variation. So we just need to be making the patient aware that they may not have a period. Anxiety, joint pain, and they could be depressed and mood changes. And we need to be aware that there could be a bone mineral density loss. Therefore, we need to be careful when you're using the higher dosage, and therefore sometimes we may have to add an estrogen like the oral contraceptive pill to counteract that. I'll come to that in a minute. Contraindication, obviously pregnancy, we can't use it, and as I said, it causes bone mineral loss density. Therefore, if somebody has got osteoporosis, we should not be using it. And if somebody has got severe hepatic impairment like Child-Pugh syndrome, then we should be using that. Or if somebody is using cyclosporine or gemcitabine, then we should not be using the medication. The most important thing is to highlight the need for contraception. because there was 49 pregnancies during the trial in a Western Caucasian population. Therefore, it highlights the need for patient counseling, that it is not a contraceptive agent. The fact that you're not having period, can make the pregnancy difficult to diagnose. And it is very important for us to counsel that it is not contraceptive and therefore you must use contraception. So the manufacturer advised using non-hormonal contraception, whereas you as clinicians can possibly use other contraception as well. So there is a trial which is ongoing with oral contraceptive pill. So logically speaking, it may be that if the fact that we have already said that endometriosis is the treatment is a hypoestrogenic state, and the elagolix acts by actually hypoestrogenic state. And if you add oral contraceptive pill, does it increase the estrogen? Does it reduce the efficacy? We don't have the data for it, but the trials are ongoing. But whereas you can certainly progestins, or like the progesterone only pill, I can't see the reason why one cannot use it. And actually, in fact, it may be beneficial, additive effect, because elagolix is working on one way and the progestin would probably add to the effect. So logically speaking, combining the two may be a good option, because I tend to do that if a lot of women who are actually medically resistant, or they're not have some improvement, then I tend to use the Mirena and the progesterone only pill together. The same logic, I'm sure we can use it here, although there is no data, but sometimes our clinical experience also can be useful, whilst we would always welcome a randomized trial data. The data may not be available, but I've certainly had sort of improved symptoms with adding two medications together. Downsides. Yes, everything glitters, new drugs, everything comes with the flashy headlines, but main problem is expensive. It is very expensive even in the US, almost up to $1,000 a month. And unless they have insurance, it could be very expensive. I suspect that is the reason it has not been introduced to a wider national health programs, such as in Europe, including UK. It is not available in our National Health Service, just simply maybe because of the cost. And as I said in the initial trial, there was no comparison with the existing treatment. Like we have got oral contraceptive pill, progesterone only pill, and already my colleagues have talked about the Dynagest and other methods which are already existent. And there is no head-to-head trial so far comparing these, but in the future, obviously, there could be trials which may ongo. And as I said, downside is it's not available in many countries, and as I understand, it's available in Bangladesh, but I don't know how many of you are using regularly, and what's your feeling on that. So now people have alluded endometriosis is a long-term condition. So therefore, if the liver function is normal, we can continue up to 24 months in 150 once daily, or up to 6 months with 200 milligram twice daily. Therefore, we can just give it up to that time and then see how things go. If the symptoms come back, then we'll have to probably reconsider the options, because we know that this is a chronic conditions until they go through the menopause. So therefore, it may be a long time before they have a good symptom control. Therefore, we may have to use repeated courses in different time. And we don't have the data on add-back therapy, like to prevent the bone loss, mineral density. But then I don't know your experience. I tend to use quite experience with the GnRH agonist injections, and we tend to give them more than 6 months with an add-back therapy with the HRT or oral contraceptive pill or Tibolone. in those women, my experience has been it's quite effective, and I don't see why we cannot do this. And although there is no evidence so far because it's a new drug. So this is one chart which may be useful because I think we go through quite a bit of data, but ultimately we are all clinicians and we need to have a clinical data to guide us our practice. So one way to look at it would be that this chart by Italian group is quite useful. So if somebody has got endometriosis pain symptom, then we say that if there is a severe features, like if they have got a urethral hydronephrosis, or bowel endometriosis with occlusive symptoms, or if you're not sure about this ovarian masses and you want to know for a histological diagnosis, you may have to do a surgical management, or if the endometrium is more than 5 centimeters, you may want to go down the route of surgery. Whereas all the superficial or any deep infiltrating endometriosis with they've got higher risk, then you may want to consider the oral contraceptive pill, or on a cyclical manner followed by continuous manner. And you can also consider the norethistron. If that is not working, then you can consider the Dynagest, which was talked about. And if there is no efficacy, then there may be a role for GnRH agonist. So it's not that, you know, elagolix is a wonderful drug. I mean, as it's an so what I would conclude is that it's actually a welcome addition to a treatment option in endometriosis when we didn't have for more than a decade a newer treatment option, because women health seems to be a neglected area where the research isn't happening, but it's a new welcome addition. But we need more information, trials, or cost reduction to optimize the treatment benefit. So I conclude that. Thank you for your kind invitation. And I would like to particularly thank Dr. Sharmeen Abbasi for all the help for the last month. She must be a superwoman who's always running around, and I'm impressed by her energy. And thank you all.
Thank you, sir. Thank you very much for enlightening us in details about the elagolix. Though we are running behind time, we are going to take 3 questions for each 3 speakers. If I'm not wrong, I can't see Dr. Mori. I'm taking the second question to Professor Juhi. Are you there? Professor Juhi, are you there?
Yes.
There's one question here. Can Dynagest be given to adolescent in case of endometriosis? And if so, how long?
Yeah, it can be given in adolescent girls, and it can be given as long as 5 years. It has been proven, the trials have been done. So it can be given for as long as 5 years. Minimum for 3 months and as long as 5 years.
Okay, madam. Another very simple question and practical day-to-day life question, that is, patients when they are on Dynagest, there is a common complaint of irregular spotting. How to manage that?
Actually, I've talked that in my lecture also, that counseling plays a very important role. Initially, there is a time where there is irregular menses and spotting, but later it turns into amenorrhea. So you have to do counseling. Counseling is very important.
Thank you,
madam. That is definitely a side effect of Dynagest. Yes, it is there.
Thank you very much. So to question to our third speaker, sir, what is the price difference between the injectable and oral GnRH antagonist?
Can I just ask, is it available in Bangladesh now, or just
No.
or is it, are you guys
No.
okay? Yeah. So for just to answer your question, the trials in the US have suggested that its costs are equal, and I suggested in my talk, actually in the UK it is not available. So you have actually the injectable one currently. And injectable one isn't that expensive in the UK National Health Service, but it all obviously involves cost of nurse administering and patient having to come. So I think it will be a significant difference when it's introduced in the UK. The reason being the ABYT has got a license on it, and they will get the money in the first years, 10 years, particularly in the National Health Services. But I was very pleased to see that actually it's about $1,000 in US in Bangladesh, it is about 2,000 taka. Is that the Bangladeshi currency? Yeah. So realistically, there's not much cost difference. But in US they say that it's probably more cost effective, but it's something we will not be able to confirm one way or the other.
So, sir, what will be your experience or what will be your recommendation, whether to go for the oral or injectable?
Ultimately, it will be the patient choice, isn't it? You will have to talk to the woman, and somebody may prefer actually, I may want to have an injection and I don't have to take 3 months. So particularly if somebody, and particularly at your setup, if it is something you can administer easily, probably it may be less costly in your setting. I would probably think the injectable would be less costly because it's been around for a long time. So if a woman would prefer oral, then potentially yes. So I don't think there is any right or wrong. Ultimately, as per everything in now currently, we have to put women at the center of our discussion. Ultimately, they choose based on our information we give it to them, and they maybe it cost, maybe they may not like injections, they'll say oral. So ultimately what I would say is that we need to put women at the center of our decision making. Thank you.
Dr. Noshaba, I just want to add one thing. In adolescent girls, actually it is diagnosis which helps us in diagnosis as well. Many times young girls come to us with dysmenorrhea and severe depletion in quality of life. You can give a short trial of diagnosis and thus you can diagnose her endometriosis as well. If she gets relief, then that means that is endometriosis. So that is one thing.
Thank you very much. Thank you very much for the information, madam. Now we have the now we are going to have the chairman's remark. May I now request our first chairperson to give her thoughts about our first speaker's presentation.
Thank you, Noshava. First of all, I would like to thank the organizer for arranging such a wonderful and informative symposium on endometriosis, the targeted medical management on endometriosis. And I would like to congratulate all the 3 speakers for their very informative presentation. And the first presentation was on aromatase as a target to treating endometriosis, and it is from Taisoka Mori. And I would like to thank him because of this informative presentation, because he explained very elaborately the molecular level, how the aromatase act in genesis of endometriosis, and why we would need to use the aromatase inhibitor as a medical management of endometriosis. There are some question also that might arise, that how long we could use, whether we can combine it with the progestin or not, and when to start, and at what level of size, at what size of endometriosis we could start the treatment, and how long you need to continue the treatment, and what benefit you could get after finishing the treatment, whether the ovulation or the fertility status improve or not. But and at the same time, the aromatase inhibitor, when we combine it with the progesterone, and then it stops the progression of the disease, and whether it stops only the progression of disease or it regresses the actual cyst size, these are the questions that we can discuss. I think the speaker, is he available?
No.
No, he's not
madam.
available. So is anybody like to add anything about this aromatase treatment on endometriosis? Will be helpful. Can the other 2 speakers, I'm requesting the other 2 speakers, can you add something about this this problem, about this question or query? Just in short.
Madam, I have no experience regarding, I have not yet used aromatase inhibitor in my practice. So I think sir will be a better person to answer this question.
Okay, I'm requesting Sanjay, yes.
Yeah, it's not used in the UK, so I'm sorry that we don't have experience with that. So obviously UK is highly regulated. So unless it goes to the proper trials, it is in the trial setting in Edinburgh, but it's not in a widespread practice. So I'm sorry, you can't comment the practical experience of that.
So there are some literature, but the literature that they showed that there's increased pain relief. And it suppresses the progress of the disease when we combine it with the progesterone. Rather than using along the progesterone, we can use this too. Okay. So thank you very much, the first speaker, Professor Daisuke Mori, for the beautiful presentation. Thank you.
Now, may I request our second chairperson, Professor Deepi Borua, to give her thoughts about our second speaker, Professor Juhi Agarwal.
Unmute. Deepi, unmute call.
Thank you, Dr. Naushaba, for your nice introduction. First of all, I would like to give thanks to the organizers, especially Professor Samina Chaudhuri, President EASB, and Professor Saleha Begum Chaudhuri, Secretary General EASB, and Dr. Sharmin Abbasi, Scientific Secretary EASB, for giving me the opportunity to chair the session. I want to congratulate all the three speakers. Thank you, Dr. Juhi Agarwal, for your excellent presentation regarding the role of diagnosis in the treatment of endometriosis. You have mentioned very nicely and elaborately, diagnosis is a selective progesterone that has anti-proliferative effect on the endometrial cells. It inhibits cytokine secretion and decreases the nerve fiber density in endometrium to relieve the pain. It has less side effects with no androgenic and no glucocorticoid and no mineralocorticoid activity. It is good. It has good compliance. You have already mentioned that compliance is 98%. And patient satisfaction is 88.9%. There is some side effects like irregular bleeding. You have mentioned nicely that improves within time. With the use of the time, it becomes regular, and that means patient become amniotic. It is contraindicated in undiagnosed vaginal bleeding, and also I want to add 2 points. In pregnancy, it is also contraindicated, and also in lactation, it is contraindicated. Diagnosis, it is not a contraceptive, and so use of non-hormonal method is recommended while taking diagnosis. You have mentioned the menstruation resumes very short time, most probably within 2 months after stopping the drug. Endometriosis is a challenging medical condition with debilitating effects on the life of the patients. So it is a potential good medical option in the treatment of endometriosis. Again, I want to give my heartfelt gratitude and thanks to Dr. Juhi Agarwal for your excellent talk. Thank you again.
Thank you, Madam.
Dr. I'm highly obliged that you have noticed minutely all my points and you have heard my lecture so sincerely. I'm really thankful.
Thank you. Thank you, Dr.
Thank you, Madam. May I now again invite Professor Salma Rauf, Madam, to give her thoughts about the third speaker.
So I'd like to congratulate Sanjay Kulkar for giving us a very nice presentation of one of the promising oral GnRH antagonist. This is a very new thing in the management of endometriosis. Hopefully, it will help the patients patient and also the clinician to treat endometriosis medically, because we want to treat the disease medically. And this is the first line of treatment, and this helps in ovarian reserve preservation. So I think it will be helpful. but it's a very new drug, and you have mentioned very comprehensively. And so far I say that's targeted presentation, that how this drug acts and what are the indication and what are the side effects of this drug. And you have mentioned that so far the comparison is not yet available in the literature. What are the added benefit of this drug over the injectable form? But one added benefit is that patient could take herself orally. That might be helpful. But the other things, that is quite expensive drug, and we don't know the long-term side effects of the drug. So as the drug is available in our countries, we must explore the opportunity and the use of this drug in patients with endometriosis, and also the price and expense also. So thank you, Sanjay Kolkar, for beautiful presentation, and we'd like to hear from you and all the speakers in future also. Thank you very much.
Thank you, madam, for your words. Thank you, everyone, for the interactive session. Now over to Dr. Utpala Majumdar.
Dear audience, we are at the end of the session. It's my pleasure to give thanks to all the honorable chairpersons, and I want to give thanks and express my gratitude to the speakers for their excellent deliberation, which has enriched our knowledge enormously. I want to give thanks to our moderator for excellent moderation. Our next session is policy session in Hall A, and which will start at 3 p.m. Thank you all for staying with us. Thank you.
Thank you. Give a big clap